everybody. Good morning and good evening in Romania. We will start as the starting hours arrived, and probably we will wait for other attendants to connect with us during our symposium. So for those that do not know me, I'm Cosmin Dumitrescu, and I'm going to be your host during the works of the symposium today. And welcome everybody to the Medical Science and Biotechnology, Romanian-American Scientists Beyond the Limit of Innovation for Curing the Incurable online symposium. This is the second conference in a series of 5 that the Consulate General of Romania in Los Angeles, together with partners from the civil society, with universities, and with other institutions from Romania and the United States, organized in order to mark the anniversary of the 10th year since the signing of the joint declaration for the implementation of the strategic partnership for the 21st century between the United States of America and Romania. So welcome again. And I would like to thank to each and every one of you for taking our invitation, and to transmit many thanks and special thanks for our partner in organizing this one for Renasterea Foundation from Romania. We appreciate what you're doing there, and we appreciate your support in organizing this conference. And last but not least, I would like to thank to the amazing scientific and academic Romanian-American community here in the United States. You're doing a wonderful job, and without you, we could not organize these wonderful events. So, as the strategic partnership between USA and Romania is one of the 3 main pillars of the foreign policy of our country, together with our activity in NATO and the European Union, we do have and appreciate of having the support of the highest political level in Romania. Last month, for the space sciences, we had official messages from the Minister of Foreign Affairs, Ambassador Bogdan Aurescu, and from the Minister of Research, Mr. Telemann. Today, we have the honor of having with us the presidential advisor and the head of the Department of Public Health from the presidential administration, Honorable Diana Păun. I would like to make an introduction of Honorable Diana Păun. So Dr. Diana Păun is a graduate of the Faculty of Medicine Carol Davila, University of Medicine and Pharmacy from Bucharest, holds a PhD in medicine from the same university, and also is an associate professor at the same university at the Department of Endocrinology. Dr. Păun holds a certificate in complementary studies in healthcare management and a master's degree in management of public health and medical services. She is a member of a lot of national and international associations, and I would like to highlight here Dr. Păun as a fellow of the American College of Endocrinology. She is the head of the Department of Public Health Administration, Presidential Administration since June the 1st, 2005. Thank you, Dr. Păun, for accepting our invitation. Thank you for joining us today. We know that you have a very busy schedule coordinating the efforts in fighting the COVID-19 pandemic, and we really appreciate your service there and everything that you're doing. You have the floor.
Thank you, Your Excellency, for your kind presentation. Good evening to everybody, esteemed colleagues, ladies and gentlemen. I'm honored and delighted to address you today on this milestone moment for our bilateral relationship, the 10th anniversary of the signing of the joint declaration for the implementation of the strategic partnership for the 21st century between Romania and the United States of America. At this welcome occasion, It is no novelty to affirm that our countries enjoy strong relations enriched by trust and friendship, which derive from mutual understanding for our shared belief that democracy is the core basis for political legitimacy and therefore stability. The sound Romanian-American community is a long-term and impactful asset to deepen our partnership to the benefit of both nations by expanding our cooperation across a broad spectrum of mutual priorities. And today's high-level forum is an eloquent example of this potential. Unfortunately, the past 14 months have been challenging for every country, every society, and every individual. Today, more than a year into a historic pandemic, there is a general international consensus that health is a crucial foundation of any society. The acknowledgement of the importance of health carries an opportunity when we start rebuilding systems that prove to be unequal and fragile. The opportunity to build back better, rethinking health, and addressing the conditions and broken systems that allow this pandemic to happen and aggressively advance. I avail myself of this opportunity to convey a heartfelt gratitude to all my colleagues, to all health workers for serving on the front line of this pandemic. They have confirmed beyond the call of duty to keep our communities safe. Innovation lays at the core of the pandemic response. The window of opportunity we now have with the vaccine rollout is the merit of innovative research and biotech. We experience the same steadfast progress driven by innovative therapeutics in treating the incurables. During the tumult of this COVID-19, we are on the onset of a new era in healthcare that, for the first time, rightfully placed the individual at its core. Personalized medicine, with the deployment of artificial intelligence and new technologies, is yielding new insights in various areas such as oncology, rare disease, or cardiovascular illnesses, delivering both clinical and economic benefit for patients and societies. Solidarity is key in post-pandemic recovery. At the same time, a strong partnership between regulators, academia, the researchers, as well as the civil society is a precondition to our quest to ensuring universal and equitable access to quality healthcare. On this solid belief, I'm confident that we are able to develop a firm collaboration in areas of interest and to build networks in new representative domains such as health, medical science, health security, and pandemic preparedness. So I'm looking forward to opportunities of working together for common well-being, and I wish you the best of success in the working of this forum. Thank you, Cosmin.
Thank you, Diana. Thank you, Honorable Presidential Advisor. We really appreciate your presence with us now. We know that it's really difficult to find time in your schedule, and we highly appreciate that. And we also appreciate your remarks, and especially those that are connected to solidarity and how everybody tried to cope and to face the challenge with the global pandemics, and to try to put together not only the public health or the global health security, but also to keep up the economies and to try to avoid all the other economic and security challenges that we, especially the democratic world, faces. Thank you for what you're doing, and we wish you have the best success in everything you're doing for managing this crisis that nobody thought that we will be faced. Thank you. So we will continue with the next speaker. As a matter of fact, is also a representative of the presidential administration. And as the Consulate General of Romania built these conferences together with the representatives of the Romanian-American academic communities here on the West Coast, we are very honored to have with us also the state advisor of the Romanian president for the relations with the members of the Romanian communities abroad. Sandra Pralong, a long-time friend and partner of the Romanian diplomatic missions and consular offices, is with us today from Bucharest, completing the team from the presidential administration. Sandra Pralong is also a PhD in political sciences, and she also completed her studies in Switzerland and United States of America. She lived in the US for a long time and came back to Romania and continued to be very active in a social life, civil society, making a great job in putting together energies and synergies of organizations in Romania and building partnerships with other organizations, NGOs abroad, especially Romanian ones. So thank you, Sandra, for being with us today, and you have the floor.
I want to welcome everyone and to congratulate the Consulate General and you personally, Cosmin, for the initiative, and also your partners, Mihaela, Joanna, and everyone else involved. It's really very much along the vision of the President of Romania, Klaus Iohannis, to have this kind of partnership between Romanians abroad and Romanians who stayed in the country in various domains of activity and actually of excellence right now. The vision is that we actually, the diaspora is not just a sad loss for Romania, but also a gain in many ways, because the experiences and the expertise that you all have gained abroad are now very much needed and welcome in Romania. And you don't have to return physically necessarily to the mother country to contribute. You can turn just your gaze back at home and contribute. And we're very appreciative of the effort that you all do. Science is an international language, a very competitive international language, and we're extremely proud of your prowesses and of your excellence in your fields, and we're very much appreciative also of your willingness to build partnerships with colleagues in Romania. So I always have this vision, you know, that I'm reminded of, that in 1848, Romania was transformed by what we call the 48ers, the Pașoptiști. And the they were only, let me remind ourselves that they were only 100, fewer than 100 people who radically transformed the country from a feudal to a modern state. They transformed everything from clothing to the alphabet to legislation, everything, just because they brought back from abroad all the know-how and the excitement of a new vision that they thus gained. And I consider that we all, I lived abroad, as Cosmin kindly reminded us, that I lived abroad, so I did the reverse migration in 1990 precisely in order to contribute to my country, and I'm extremely impressed and honored is too weak a word. I'm really humbled by all the expertise that you all hold and are willing to share with your colleagues in Romania. So thank you very much. I'm going to listen to as many of the presentations as I can, but I have a baby of 95 whom I have at some point to put to bed and then return. So I also, just one other remark, I'm hearing very broken signal. I'm actually not in Bucharest, I'm on the seaside. It's too dark outside to show you the beautiful Black Sea. And I have a very bad signal. So at some point, if it gets too choppy, I'll cut off the video. So don't think that I disappeared. Thank you.
Thank you, Honorable State Advisor. Thank you, Sandra, dear friend. We appreciate your remarks and your kind words about the members of the Romanian communities abroad in general, and of course, especially thinking about the Romanian-American community that is so active and so involved in communicating with the mother country, as you called it, with Romania, and also very interested in developing direct connections and relations with researchers in Romania, Romanian universities, and generally speaking, with this environment of scientists that take our world forward. So thank you for your kind words, and I'm kind of take advantage of your remarks to make the connection to what we are facing right now and what we learned in the last 14 months, as Diana said before. So with this global risky COVID-19 pandemic, we learned a lot of lessons. That when we learned first that nobody is prepared enough to face such a challenge, and that solidarity, national and international one, is very important, and we should all act together in order to be successful against such danger, such threat for our global security. And we learned also that the health security is as important as any other concept of security, and that we should cherish and we should cultivate our scientists in order to be proficient and successful in fighting that. So that is why one of the objectives of the Consulate General of Romania in Los Angeles with our partners, and this time I would like to value the contribution of Renasterea Foundation in Romania, that agreed in cooperating with us in order to build or to consolidate these kind of relations. Of course, that we always think about bringing together all energies, not only science, not only administration, but also to bring with us the civil society. And who else is a better representative of civil society than the NGOs that work with people, that try to explain to people what the administration is doing, what the scientists are communicating, what's the purpose of science, and what's the benefit of science and administration together for the national and international health security. And we are very blessed to have with us today the president of the Renasterea Foundation. They are doing a wonderful job in Romania in explaining people and educating people, and not only in Romania, but also in Europe, as far as I know. But I'm looking forward to the presentation of Mrs. Mihaela Joana, the president of the foundation. So we'll dive into the subject of our symposium today with our first speaker, and Mihaela Joana, the president of Renasterea Foundation, that in September 2001 established this foundation, an NGO aimed to raise awareness about the importance of early detection of women cancers. In order to increase survival rates in Romania, since 2003, she is the national representative of Europa Donna in Romania. Since 2007, Renasterea Foundation organized and implemented the first pilot screening program for breast and cervical cancers in Bucharest and in rural areas through Renasterea mobile diagnostic units with over 30,000 patients tested. This project represents the basis of the national screening program that is to be implemented in Romania by the Ministry of Health. Between 2007 and 2015, she was the president of the Romanian Red Cross. During her tenure, Mihaela Joana initiated national projects, Food Bank, National First Aid Network aimed to restore the concept of volunteering and philanthropy in Romania. Due to her efforts, the topic of breast cancer was included in the Romanian Parliament agenda on 2014, October 1st, and was recognized as a national day of fight against breast cancer. Since 2018, Mihaela Joana is the president of Think Pink Europe, a European NGO that aims to bridge the gap between healthcare systems in European countries with the purpose of breast and cervical cancer prevention, diagnosis, and medical care. She has put together medical specialists, actors from patients' organizations, Romanian and EU parliamentarians, organizing debates and roundtables on improving prevention, access to healthcare, and the quality of medical services in Romania. Thank you, Mihaela, for being with us.
Thank you very much. Thank you. That's a great presentation. I'm impressed. We're honored as a Renasterea team to be part of this great event organized by the General Council. Thank you very much, Cosmin. I'd like to thank also the representatives of the Presidential Administration and every participant to our event. My presentation will be very short as some of it was already presented by Cosmin. Renasterea Foundation was started also with the model of US organizations like Evelyn Lauder Foundation and Susan G. Komen Foundation, organizations that I worked with while I lived in the US between 96 and 2001. So in 2001, upon returning to Bucharest, I established this foundation. What we did is we concentrated on organizing awareness campaigns. The difference between Romania and the US in 2001 was huge in terms of survival rates. And these differences were mostly due to lack of awareness, to a different mentality, and the lack of national screening programs. And I, together with a few close friends, thought that we can do something about that. So we developed awareness campaigns like the pink illumination events, galas to raise funds, but also to provide this access to tests to women who of low income or who encounter different barriers to an early diagnostic. We did build 2 medical centers in Bucharest, 2 mobile units that provide free of charge tests, and the numbers of tests we provided are on the right. The pink illumination event has been held every year at 1st of October for 20 years. And this is just a nice way to remind women that another year has passed and they need to do their checkups. It has been very successful. Our medical center of excellence was built in 2011, and it's a very modern, well-equipped center, an investment of 2.3 million euros, partially like 80%, which is more than partially financed through a grant from European development funds. The mobile unit, actually we operate 2 mobile units. We purchased them with funds we raised through our events. And these are the results that we had. More than 22,000 women were tested for, with mammography and pap smear tests for free in rural locations. I would mention here that all the projects of Renasterea foundations were developed with private funding or European funding or Norwegian funds. We never asked or accessed Romanian budgets funds. Because in European Union, women live or die depending on the country they live in. And these are numbers that come out of professional studies. A woman that develops breast cancer, for example, in Eastern European countries have a 15% lower chance of survival. And this is why we started Think Pink Europe. Think Pink Europe is an European organization. It's geographic Europe, I will tell you in a second, and its purpose is closing the gap in the medical care, in access to the timely diagnosis and modern treatment. Think Pink Europe was set up in 2018 at the European Parliament. It started with 5 countries. And today we have NGOs from 32 European countries that are members. I am the chair of this organization for the time being. One of the main events organized by all these 32 organizations is Race for the Cure. This is, again, a project that started in the US by Susan Komen Foundation. And Think Pink Europe has a partnership with Susan Komen Foundation in organizing Race for the Cure. It is Europe's biggest sporting event for women's health. And this is an image from Roma Race, which hosts more than 70,000 people for a 5K race. Romania did that for 5 years. And last year we had an online event. Last year we did held an online event, and it's great because more than 5,000 people in Romania just went around their house with friends and posted their participation. These are some numbers from ISRA study about survival rates in Romania versus Europe and Belgium. As you can see, if in Romania we have a 64% rate of survival from women's cancers, in Belgium they have an 84% survival rate. It's 20% more women live, if they live in Belgium, compared to Romania. We sponsored a study done by Deloitte, and they have important conclusions in the terms of the costs of breast cancer, for example. And only 30% are direct costs, and they represent 0.16% of Romania's GDP in 2017, and 70% are indirect costs, like medical leave, social assistance for disability, loss of productivity, family and caregivers costs. These are very interesting conclusions, and I would suggest that we share these presentations so that we can take a look at the details that are interested for each of us. So our purpose is closing the gap. We demonstrated in a nutshell in pilot projects that we can organize screening campaigns, access for rural population to diagnosis. We did 3 years of campaign, and we sponsored 2 studies together with our sponsors, of course. And we got 2 government pledges. So we can prove that we can work with the authorities, and we can improve the patient's chances of survival. So the 2 pledges of the government are to implement a national breast cancer screening program, which is starting being implemented first regionally this year, and we hope it will become national, and to continue the national cervical cancer screening program that has started, and we hope to have more women attend it. There are 3 EU-funded programs with regional screening programs that have started, as I was telling you. And what is important, I think, for Romania would be to leapfrog in order to close this gap, to adopt newest technologies like telemedicine, personalized diagnosis and treatment, access to the modern treatments, and share research and best practices. One other part of the activity, which is very important, is to convince and implicate the members of the parliament in the fight against cancer. And we know that the US Congress has a cancer caucus. It has 40 members, and it's bipartisan. The European Parliament has members of European Parliament Against Cancer group. It was founded in 2005, and just in 2009, the Romanian Parliament established a cancer caucus, which is multi-partisan and bicameral. And the Aspen Institute has an initiative to bring together these 3 caucuses in programs to share best practices. European Parliament has, and the European Commission launched the Europe's Beating Cancer Plan 2021-2030. And I think this is a huge opportunity for Romania, as the EU budget has earmarked $4 billion to address cancer. And these are the pillars of this European Cancer Plan. I think these are opportunities not to be missed. And we all have a role to play: NGOs, administration, parliament, of course, the Ministry of Health, mass media, everybody, researchers, and of course, Romanians living abroad. Thank you very much. Together we're stronger, better, and able to close the healthcare gap. Thank you.
Thank you for most interesting presentation, and congratulations for your accomplishments as a foundation and personally. We can see how diplomacy expands.
Of course.
And how
I started in Washington,
yes.
and I was inspired, but by what is happening here, and I was able to bring that to Romania, you know, being untainted by political influence and just building together with everybody these projects. Thank you very much.
Thank you. And we can see how these segments of diplomacy, the parliament diplomacy, parliamentarian diplomacy, the classic diplomacy, and the activity of NGOs come together in order to form a synergic act and to bring forward, not one step, but many steps, our cooperation between Romania and the United States. And the model of cooperation of the American Congress it's exceptionally wonderful. So, and we have a lot to learn from that, and bringing together these caucuses, I'm sure that it's going to be a win-win situation for everybody. And also, we appreciate what you are doing with your organization for educating people. And we see now that all the efforts that all the states are doing for teaching people, educating people about vaccines. We all know that the way back to normality is to have successful vaccinations campaigns. And the role of media, of mass media, and of the NGOs is of utmost importance in educating people, explaining people the benefits of getting vaccinated in order to fight this COVID-19 pandemic. I wish you all the best success in everything you're doing. And also, if we are talking about education, we pass on to the next speaker. We'll stay in Romania, and we have the honor of having with us the rector of a University of Medicine in Romania that is growing very fast and with amazing results in academic area, but also in medicine, clinical medicine. It is my honor to introduce to you Professor Dr. Leonard Azanfirei. So Professor Leonard Azanfirei is an MD and PhD, director of George Emil Palade University of Medicine, Pharmacy, Science and Technology of Târgu Mureș, professor of anesthesia and intensive care at the same university. He is the editor-in-chief of the Journal of Critical Care Medicine. Professor Azanfirei promoted the university accelerated development by implementing modern training methods, including medical simulation, the use of virtual reality in medical training, as well as the digitization of the educational process. In 2019, he founded in Hamburg, Germany, the first medical campus of a Romanian medical university in a foreign country, and enforced a strong university internationalization by including it in numerous international projects and research networks. He initiated new study programs, including those of medical engineering, computer science, artificial intelligence, information technology, and bioinformatics. He holds the title of Doctor Honoris Causa of medical universities in Chișinău, in Republic of Moldova, and Tbilisi, Georgia. At national level, Leonard Azanfirei is the Vice President of the National Council of Rectors of Romania and President of the Romanian Society of Applied Simulation in Medicine. At the same time, he is the Vice President of the Romanian Senate, a member of the Health Committee and Education Committee of the Romanian Senate. Thank you, Professor Azanfirei, for being with us. We are very honored to have you here as a scientist and also as a very high official. The floor is yours. Thank you.
Okay. Your Excellency, dear colleagues and guests, it's a privilege and a great responsibility for me to give a correct image of today's medical research in Romania. And thank you, Mrs. Mihaela Joana. By the way, I was impressed by your great projects. Thank you, Mr. Cosmin Dumitrescu, Consul General of Romania in Los Angeles, for this opportunity. The healthcare system in a country is a picture of the current state of affairs, while medical research shows what this system will look like in the future. In Romania, medical research has not been a priority for a long time. Although, as in any country, and even more so in Romania, medical research constantly needs the most expensive resources. Predictable funds and grants outlined to each excellence center accordingly, innovation-focused specialists, internationally connected teams, modern infrastructure, facilities, and equipment to align to the international laboratory and research. Okay, in what follows, I will try to build up a picture of the medical research in Romania by providing a set of official data and by interpreting them. In Romania, medical research is mostly conducted within the University of Medicine and Pharmacy. You most probably know that in Romania there are 6 traditional, well-established medical universities located in Bucharest, Cluj-Napoca, Iași, Timișoara, Craiova, and Târgu Mureș. And there are also a number of smaller medical schools founded after 1990 in other cities such as Brașov, Sibiu, Constanța, Oradea, Arad, and Galați, within which there is mostly medical education and less medical research. Furthermore, there are few medical research institutes and several high-performing hospitals which carry out medical research as well, and which are located in the main traditional university centers. On this map, you can see the distribution of this institution with large regions, which currently represent the development regions of Romania, so also the European founded regions. Naturally and historically, there is a concentration of resources in Bucharest. Generally, in Romania, large medical research projects have 2 major sources of finance: national public funds and European funding. Private funding for research projects is insignificant. Rationally, the number of projects is in line with the number of institutions within each region. This map shows the distribution of nationally funded projects, revealing that if you compare the number of research projects to the number of the research institution, it becomes apparent that there are significantly different rates of allotment from one region to another, with variation between 7 to 74 projects per institution. This may be the result of the fact that medical research is not necessarily a matter of infrastructure, but it mainly depends on the people involved in their international connection, and why not on the research culture developed under the time. The situation is different with regard of European Union funded projects, most of this having Romanian institutes as partners, and extremely rarely as project leaders. Obviously, in this case, the competition is extremely tough, and it also involves an important international lobby component. Most certainly, the majority of the Romanian research institutions are power deficient when it comes to enforcing their own project at European level in the medical field. It should be noted that currently in Romania, there are numerous medical research centers with infrastructure of the highest quality. Opportunity of European funding have been taken advantage of mainly by university, which have set up medical research centers, especially in fundamental research. The centers are well equipped and they invested in high performance equipment. As a result, at national level, there are 462 research infrastructure of high performance potential, though they are underused sometimes. Such an European founded infrastructure is a center for advanced medical and pharmaceutical research belonging to George Emil Palade University of Târgu Mureș. An UNESCO analysis of 2018 statistics reveals most serious problem within the Romanian medical research. There is a limited number of 882 researchers per million inhabitants in Romania. Maybe the figure is not entirely accurate, because in the Romanian system, the academic staff is also involved in research from the position of academics and not that of mere researchers. the number of researchers is decreasing. This number is the lowest in the European country in the region. For example, Ukraine has 988 researchers, Germany more than 5,000, Denmark 8,000, Bulgaria 2.3, Slovakia 2,900, Czech Republic 3,800, Russian Federation 2,700, and the average number for the euro area is 4,180, and for North America 4,400 researchers. So the small number of Romanian researchers partly explains the current state of research and originates mainly in the outrageously low public financing of Romanian research, as well as the policy of all the government of the last 30 years. Furthermore, I shall make a brief analysis of the publication by the Romanian medical researchers in important medical databases, Web of Science core collection. When it comes to the entire scientific production of researchers in Romania, I'm content to notice that there is a national database, the name is BrainMap, which provides an up-to-date situation of the entire scientific performance. Surprisingly, between 2016 and 2018, the average number of published articles per researcher is notably large, 29.8, being second only to Poland's, although we have the lowest number of researchers in Europe. We must understand the explanation for this paradoxical situation. Another aspect of note is the high percentage, 27%, of scientific articles related to medicine published in 2020, compared to 17% for the 1975 to 2019 period, especially when it comes to articles published in scientific journals, 31%, and books, 36%. Adding 2020 and 2021, we obtained almost 51,000 articles published in a Web of Science core collection, out of which only 37% published in highly cited in field, hot papers in field, and open access category. With reference to these articles, quantitatively speaking, the peak number of publication, that is 4,700 items, was reached in 2019, followed by a small decrease in 2020. So the trend seems to have been favorable by 2020. According to the Web of Science classification, the main, wait a second, I have to go back. I don't know. I think I have to exit. Just a moment, excuse me. Yes, thank you. So according to the Web of Science classification, the main area of interest for publication were those of fundamental research are biochemistry and molecular biology, pharmacology, pharmacy, followed by internal medicine, a more general field, cardiovascular and neuroscience area. I must point out that the following image are Web of Science screenshots currently managed by Clarivate Analytics, obtaining using the analyzed results and search titles. So for Romanian researchers, it becomes clear that their chance to publish is higher in fundamental and interdisciplinary research than in clinical research. In the wider landscape of the research field addressed by Romanian researchers, the medical area has a significant weight. It's my belief that the increasing visibility of the Romanian medical research stems from the internationalization strategy that is accessing an international research network that each university has adopted. In all published articles, the authorship for Romanian authors and foreign co-authors, US is a third partnership country, following France due to our historical Francophilia, and to Germany. Unfortunately, I don't have a situation with the Romanian diaspora, but I think that here we find another part, the most valuable of Romania. As far as publication are concerned, only 2/3 of these works have exclusive authors from Romania. The remaining 1/3 being international collaboration. I must remark here something significant for this meeting, the fact that the first associated country is, again, US. It does not surprise me, and it is a reason for hope and maybe for the future. Here are the main institutions in the United States with which Romanian medical institutions have previously worked, usually as interpersonal cooperation, resulting in joint publication. Again, in many of them, there are Romanians who left Romania, just like you, who have achieved professional success there, and who have remained friends with their home country. Related or similarly, you consider the US Department of Health Human Service and the National Institute of Health together, we notice that the United States ranks first, exceeding the European Commission as founding agency for the papers published by Romanian medical authors. I can't overlook or ignore some Romanian authors' achievements as co-authors of articles published in major medical journals such as New England Journal of Medicine or Lancet. In order to have a quantitative and not just a qualitative and not just a quantitative assessment, we also must assess the journals where Romanian researchers submit for publications. Most of the journals in which the articles mentioned above were published, especially those having exclusively Romanian authorship, are also edited in Romania. And this may lead to a discussion on the relevance of quality of this undertaking. This may be a possible explanation for the considerable productivity above mentioned in comparison with a small number of researchers per 1 million inhabitants. The inclusion of Romanian journals in the Web of Science core collection is a quality mark given to these journals, but the value of the impact factors and the number of citations of the journal and of each article published on them, these are what give the real picture of research quality beyond quantitative evaluation. And beyond the passion for research done as a vocation, there is often the pressure to publish at any cost and anywhere, either to advance in the teaching career or to defend and complete the PhD thesis. Nevertheless, there may also be the matter that publication fees for many international journals are still prohibitive for many Romanian researchers. The issue of underfinancing, however, remains the main problem of research in the country. Romania holds the last position regarding research and development among the European Union countries. According to the European Statistical Office, Eurostat, in 2019, research and development expenses in the European Union accounted for 2.19% of GDP. The highest percentage of GDP allocated to research and development expenditure, above 3%, was in Sweden, 339, Austria, and Germany. And on the other hand, 8 member states allocated less than 1% of their GDP to research. And here we find another explanation of the existing situation, but also the extent of the effort made by researchers and Romanian universities to find alternative source of funding and to be part of projects with international funding. The loss of young and educated workforce is a problem which we can't sweep under the rug any longer. According to the Global Competitiveness Index, the countries of Eastern and Southern Europe are the ones who manage least to keep their valuable young people. For example, almost 3 million Romanians, especially highly qualified workers, live today in other European Union member states. The European Committee of the Regions is concerned that the phenomenon of brain drain will pose a serious risk to the long-term sustainability of the European project if the social and economic imbalances between the states exporting and those importing well-prepared young people are not resolved. The future of Romanian university is threatened not only by the overall demographic decline, but also by the constant growth of the Romanian young population who lives to study abroad and who, as a rule, are the best high school graduates, many of whom no longer return. Moreover, there is a significant drop in the number of university students in Romania. A World Bank report warns that Romania is severely affected by the brain drain phenomenon, about 40 of Romanian immigrants having graduated university. I think we can all agree these data are worrying, if not alarming. Honestly, I don't think we have too many mechanisms now to convince those who left to return. But I think it is our duty to make sure that those who have not left yet start to want to stay in Romania. To conclude, I believe that one of the solutions that we should urgently adopt is a much stronger connection with the Romanian diaspora, attracting foreign researchers to work longer in Romania research institution and be part of future research projects. Not because our researchers will not be up to the task, but because interaction between professionals must become a priority in order to strengthen Romania's position in international research. If I may, I offer to set the wheels in motion. I challenge anyone who is listening to us now and want to effectively join as a research director project at George Emil Palladiu University of Târgu Mureș, which I run, to contact me subsequently. I believe that there are opportunity. There are also funds, as many as possible, and there is a full openness. A new national competition, for example, is to be opening presently. Its name is Exploratory Research Project Competition, where diaspora researchers may well be included. You are very welcome. So at the end, research has no burden, no walls. Its purpose is to create bridge and networks, because more than other fields, the research has put Romania on the world map by exporting intelligence. Thank you.
Thank you, Professor Azanfire. A very interesting, synthetic, and at the same time comprehensive presentation about the realities that Romanian research field is facing right now. And I'm sure that not only Romania, and I'm sure because I have the necessary information, not only Romania faces that risks and threat of the brain drain in Europe. As a matter of fact, you pointed out several good things about the excellency of the Romanian medical education system, because Romanian universities are practically providers for hospitals all around the world. And we can find Romanian doctors everywhere that are really and highly appreciated for their work, not only in hospitals, but also in research teams. At the same time, you pointed out some concerning aspect that should be taken into consideration, and we should find solutions. And you also provided us a way in which this thing could be solved, attracting our amazing, wonderful representatives, Romanian representatives in international teams that have extraordinary accomplishments in research. And maybe we should learn how America is coping with the financing world of research, and how they bring private sector near the universities and find a way to finance everything. And I'm sure that with the help of specialists, as the ones that we are having today as speakers here, we will find a solution. And I'm really glad to see that also they are very interested in giving back to Romania after they were educated there. And there are a lot of them here on the West Coast. I'm very grateful to them and for what they are doing for universities in Romania, because they do have great accomplishments, as our next speaker has. So Mr. Adrian Bot, Dr. Adrian Bot, an amazing professional and the one that found with his team a cure for an incurable disease, illness. He's with us today. He accepted our invitation. He received in 2018, if I'm not wrong, he received the highest award in his field for the cure for non-Hodgkin's syndrome, but I'm leaving him to tell us about that. So allow me to introduce Dr. Adrian Bot, who is the Vice President and Global Head of the Transnational Medicine, as well as the Head of Research, Santa Monica at Kite and Gilead Company, a Gilead company developing genetically engineered cells for oncology indications, including breakthrough CAR-T cells product currently in clinical practice. Dr. Bot has 23 years of experience in biopharmaceutical industry with focus on discovery and development of immunotherapies. He performed pioneering work on two categories of transformative treatments: gene-based vaccines and T-cell therapies. He obtained his MD in Romania in 1993 and his PhD in biomedical sciences at Mount Sinai School of Medicine in New York in 1998. Subsequently, he was a guest scientist at the Scripps Research Institute in La Jolla, California. Prior to his appointment as a chief scientific officer at Kite Pharma in 2011, and then vice president and global head of translational medicine, and more recently Santa Monica head of research, Dr. Bot served in various senior R&D leadership positions at Mankind Corp and Alliance Pharmaceutical Corp in La Jolla, California. His present or prior activities and appointments include scientific advisory board and Allicin Therapeutics editorial boards at Journal of Immunology, Journal of Transnational Medicine, International Reviews or Immunology, and leadership appointments in global professional societies, as the Society of Immunotherapy of Cancer. Dr. Bot, we are very, very honored to have you with us today, and thank you for accepting our invitation and joining the works of the symposium. The floor is yours. Thank you.
First, I would like to thank you very much, Your Excellency, for the extraordinary introduction. It is truly an honor and a privilege for me to be here, and always an honor to be in presence of such luminaries in diplomacy as well as in technical fields, medicine and science. I will bring up my presentation, if that's fine now, and you let me know if we can see that. Okay.
We can see that.
And I will also bring up my laser pointer, if that's acceptable, so we can go through some of the interesting science that we developed along the years. But I would like, before starting, actually to mark this emotional moment, 10 years anniversary of the strategic partnership between the US, our adoptive country, and Romania, our native country. What an achievement, and what an extraordinary component, actually the backbone of our external policy. I'm very proud to be serving both countries. And also I was very touched and humbled by the remarks of the Honorable Dr. Paun as well as Ms. Pralong, and of course the extraordinary activities and achievements of Ms. Joanna with the Renasterea Foundation. and the aspirations that were outlined, articulated by Dr. Azamfire. And I resonate 100% with those. And anything we can do on our side, please let us know, and we'll be here to support and help out. But I will not start my presentation before trying to characterize in one single sentence His Excellency Cosmin Dumitrescu on what he's doing for our country and the relation between US and Romania. And the best way, I think, and I hope he does not mind that, to characterize those efforts, he's a man larger than life, of encyclopedic knowledge that is matched only by his energy. I still want to know what's the secret, but we'll talk about that later on. In any case, let's go to the next slide. A disclosure, I am in the senior management of Kite, a Gilead company focused on cell therapies, right now the largest global organization involved with development of cell therapies, but I also serve on boards such as the Elisio Therapeutics, bringing forward the next generation vaccines against SARS-CoV-2 as well as other threats, global threats. My passion has been always discovery and development of transformative treatments that address global threats and global needs as well as patient needs. And I will start by just sharing with you a little bit of history in terms of where treatment modalities, how treatment modalities evolved in cancer, starting on the left part of the slide with chemotherapies still in use broadly across various oncology indications, but treatments really that target both cancerous cells, and unfortunately, as a bystander effect, they also affect normal cells. So really the last 30, 40 years were about advancing precision medicine, treatment modalities that are capable actually to modify the disease process while leaving in place normal cells. And immunotherapy is a major component of this particular effort. As you see here, with the advent of monoclonal antibodies in the 90s, then going to active immunotherapy strategies to reprogram the immune system, either through biologics or small molecules, as well as more recently to gene programming of living cells, leading really to a breakthrough in medicine, opening wide actually the area of gene intervention, both ex vivo as well as in vivo. And I'll talk more about one example that actually I think helped us understand the potential as well as the limitations of gene-based intervention. But at the core of the problem here is understanding how we can utilize an immune cell, how we can reprogram it to be able to recognize and eliminate the last cancerous cell in the body, knowing that the problem that we are facing globally is cancer recurrence or metastasis. It is important for us to understand how to harness an immune cell as a precision medicine to be able to actually do that job, knowing that immune system evolved primarily to deal with viruses, in general microbial infections, but not necessarily cancer. So the way we are trying to achieve that, we and others for the last more than 1 decade now, is by reprogramming a T cell, which is a major component of the immune system, in charge of orchestrating the defense against microbial agents, such as including viruses, SARS-CoV-2 obviously is an example, as well as other broader range of immunological threats, and taking those particular T cells, the central component of our immune system, and modify them, transduce them with chimeric receptors or synthetic receptors, man-made, that enable them to sense cancerous cells and destroy them. So that's the core of the technology that I'm going to talk about today. And for us, 10 years ago, there was, when I joined Kite as a chief scientific officer, there was an important question ahead of us. Knowing that this concept has never been tested, what's the best way actually to test it, to achieve proof of concept? and show actually that a reprogrammed immune system is capable to induce tumor regression and eliminate cancerous cells from the body in a metastatic setting. And the disease indication that we turned to initially was a type of non-Hodgkin lymphoma that is very dreadful, called diffuse large B-cell lymphoma. In most serious cases, this advances rapidly without any practical effective treatment. So third stage non-Hodgkin diffuse large B-cell lymphoma was exactly the setting where we turned first our attention, as these patients usually have only weeks or at best months time to live. And how we try to achieve that? We try to leverage what United States is the best at, is creating that ecosystem in which they can rapidly identify and bring forward cutting edge research and translate that to transformative products across all fields. So the four pillars of this ecosystem are, of course, access to technology, number one. Number two, access to financing through venture capital. Number three, access to scientific talent. And number four, a strong IP, intellectual property environment. So we took advantage of this when we formed Kite almost, or more actually, 11 years ago, more than 10 years ago. And as you see on this particular diagram, chronological, let's say, list of milestones all the way to present, we were able in about 5 years, 6 years time frame since we initiated research development on a first-in-class product that I briefly described, all the way to its approval by the FDA, we were actually in a position to bring this to the patients in need in a commercial environment. In fact, in 2018, as His Excellency mentioned, this first-in-class product called Axicell received the highest distinction in the biotechnology field, back-to-back with a similar product developed by another organization. How are we able to achieve this? What are these products about? Well, these are living products. They're obtained from living T cells that are harvested or generated from each and every patient, and they're transduced with a vector, a genetic vector that achieves expression of that artificial sensor or receptor that I was talking about, the chimeric antigen receptor, able actually to help. Yes.
I'm sorry to interrupt, but I think you don't share anymore with us the presentation. We cannot see it anymore.
Okay, let me try to get back in the mode.
Apologize for interrupting, but it was necessary.
Absolutely, and I apologize. It may be a bandwidth issue on my side. Some of these slides are very large and loaded. What about now?
Yes, we can see that now. Thank you.
Thank you so much for letting me know, and I'll bring my pointer. So how are we able actually to generate this type of products, genetically engineered T cells, living cells? So the way we do that is by harvesting immune cells from patients and then reprogram them with genetic vectors ex vivo in manufacturing bags, specialized incubators, then formulating them actually and providing them back to each and every patient in need. So it's truly a personalized approach, precision medicine-based approach involving a living cell. And what do these particular cells do in the body once we infuse them? As we see here, CAT scan as well as PET scan of 2 patients. Actually, this is, I believe, both of them are chemorefractory, yes, diffuse large B-cell lymphoma patients, part of one of the trials, many trials that we conducted. And these cells, when they're infused in the body, they multiply very rapidly, and they search and destroy residual cancer cells and eliminate metastases in the body of the patient. That's how they work. And once they do the job, they perish. So they don't live for a long time, only as long as they need to actually perform their job. And if you look at registrational trial, large trials, multi-center trials, testing this particular concept, what we have here is the ZUMA-1 trial that is utilized to initially register this product. It really shows that if you follow these patients, about 100 of them a long time, you see that about 40% of these patients with life-threatening terminal disease achieve a durable, complete response. It's quite impressive in a setting such as refractory diffuse large B-cell lymphoma. These type of treatments actually are very powerful immunotherapies. They come with side effects, temporary acute side effects such as cytokine release syndrome, something that we are seeing actually in a setting in context of COVID-19 infection that can be generally manageable with corticosteroids as well as IL-6 receptor blockade. A little bit more science here, going back to the first trial of its kind initiated at the National Cancer Institute, our partners, in terms of developing this type of products. We see here that, that we have responses with this type of treatment that last over 9 years at this point, creating actually the possibility, supporting the concept that genetically reprogrammed immune cells are actually capable to eliminate the last cancerous cell in a subset of patients. My job as the head of translational medicine and discovery research is also to understand how things may go wrong, not only how things go right. So we deployed a very substantial, vast biomarkers analysis of all patients that were dosed with this type of products during the last almost 10 years. And we utilized artificial intelligence, machine learning, to deconvolute what are the major attributes of products and patient characteristics that support actually or lead to complete cure versus treatment resistance. And this is extremely important because this type of information will allow us to design next generation approaches that are more effective across broader categories of patients. Very rapidly, we identified product attributes dealing with the immune system, tumor characteristics, and key elements of the antigen biology that are important to achieve a response through either affecting or influencing CAR T cell expansion in vivo, as well as independently affecting durable response versus treatment failure. And all of these being connected to the pre-treatment state of the immune system on each and every one of us. This type of information really leads to key ways, actionable strategies on how to improve the efficacy and diminish the toxicities of these new generation of products. Where we are right now, we are at the end of the beginning of this revolution of gene programmed T cells for cancer, which started with the approval of 2 products, breakthrough products in 2017. And to date, we have 5 products approved across a broad range of malignancies in this space, and this revolution continues. What is really next for us in order to maximize the footprint of these precision medicine-based products? Of course, to expand the applicability to a variety of different diseases, oncology and non-oncology diseases alike, to increase their efficacy, reduce their toxicity, as well as to improve the access globally by making medicines that are cheaper and can be deployed as off-the-shelf medicine across the globe. And I'm going to leave you with this picture. What's the role of immune engineering in the modern medicine? And I feel very strong about the concept of reprogramming the immune system ex vivo or in vivo in order to treat a broad range of disorders. But all of this started with very modest efforts back decades ago on what we call DNA vaccines or somatic gene transfer, to reprogram antigen presenting cells and create a new generation of vaccines. But where we are now, we have two categories of transformative products in this space, already helping millions and millions of patients in need. We have genetically engineered T-cell products, 5 approved to date in cancer, as we discussed. And we also have the concept of RNA-based vaccination, which is similar in somatic gene transfer, achieving transient expression of the spike protein in antigen-presenting cells, leading to strong T-cell and B-cell responses. So how these concepts will converge later on, I believe, through actually bringing forward technologies to in vivo reprogram the immune cells with applicability across both regenerative medicine, inflammation, and oncology. With this, I would like to finish my presentation, not before I acknowledge the whole actually research and development organization at KITE. I've been privileged to be part of it and to lead many of the activities during the last 10 years. I would like also, of course, to acknowledge all our network of principal investigators from the key comprehensive cancer centers and immunology centers, especially in the United States, but across the globe as well, as these type of medicines have been approved in European Union and beyond. I would like also, of course, to acknowledge the patients as well as the caregivers, and special acknowledgements to my wife, Dr. Simona Bond, who is actually on the call today for being my partner and support for the last almost 3 decades. And I cannot finish without sharing with you 2 beautiful pictures from my native village in Banat, Romania, Teregova, Karaș-Severin. And with all of this, again, many thanks for your attention, and I'm looking forward to questions. Thank you very much.
Thank you very much, Dr. Bot. Even if I had an idea about the substance of your research and the achievements that you had together with your team, I must confess that it's absolutely fascinating to learn again and to see some details of your work. Congratulations for your achievements and best success in everything you're doing in the future. You mentioned that already millions of people are benefiting from the results of your research, and I'm sure that this technology that now we all became aware of with the COVID-19 vaccination and with the RNA messenger vaccines is a big part of a future in medicine. Thank you again for your presentation, and I throw the glove of the challenge of questions at the end of the presentation in the Q&A section of our symposium. And it's good that you mentioned so many times in your presentation the applicability of the results of your research that is fighting cancer, because let's face it, during the last 14 months after the outburst of the COVID-19 pandemic, the main causes for death, for fatalities from illnesses was not the COVID-19, but the cancer and the cardiovascular illnesses.
It's the cytokine release syndrome, and that's something that actually we learned a lot from bringing forward potent immunotherapies, as Dr. Radu, for example, knows very well, and elucidating the key mechanisms of why this excessive inflammation occurs in context of viral infections and immunotherapy allowed us to bring forward novel strategies to tackle that, including IL-6 receptor blockade and even low-dose steroids, which I was very glad that was implemented successfully in Romania, and congratulations to all for that. But I would like also to outline that in this day and age, it is about teams, highly functioning teams. It's not about individual researchers, but teams coming together with their strengths and synergies to make these things happen. So I just wanted to make sure that that's a concept that is very widely, publicized and shared. Thank you.
Thank you again for your last remarks. Yes, team working is the solution, and it always brought the best results. And I was mentioning that the main causes of death, of fatalities in the hospitals globally, the causes are the cancer and the cardiovascular diseases. And I was doing that for making the connection with our next speaker, that is a clinician. So it's world-known and renowned surgeon, heart surgeon, Dr. Alexandru Mărmureanu. We are very lucky to have him with us today. Usually he is in the OR. It's very rare occasions when you can find Dr. Mărmureanu outside of the operation room. And we are so lucky to have him with us today. He is going to give us a presentation about minimally invasive cardiothoracic surgery. Alexandru is also a good friend and an amazing person outside the OR. Thank you, Dr. Mărmureanu, for accepting our invitation, and allow me to say several words about his career for introducing him. So Dr. Alexandru Mărmureanu is a prominent board-certified thoracic and cardiovascular surgeon with special expertise in the field of minimally invasive surgery, where he pioneered and developed several techniques and procedures. In his 30 years of practice, he has been able to successfully treat some of the most complex and challenging heart and lung conditions with high rates of success. Dr. Mărmureanu completed his general surgery residency and research fellowship at the New York University Medical Center, NYU, in New York City. He continued his surgical training at the Mount Sinai Medical Center in New York City, where he was actively involved in the field of thoracic and cardiovascular surgery research. He completed his cardiothoracic surgery fellowship at the University of California, Los Angeles, UCLA, in 2002. He continued to practice as UCLA faculty before joining Cedar Sinai Medical Center and also becoming a director of several cardiothoracic surgery programs in LA. Dr. Mărmureanu is nationally and internationally recognized for the field of thoracic and cardiovascular surgery and has authored numerous research papers and clinical publications. Active both in local and international charitable causes, Alexandru donates his time for traveling around the world to train local surgeons and perform pro bono procedures. He has appeared as a guest speaker in numerous TV and radio shows where he discussed the benefits of cutting-edge surgical techniques, as well as advantages of minimally invasive surgery for thoracic and cardiovascular patients. Dr. Mărmureanu maintains a busy and active cardiothoracic surgery practice in several hospitals, where he performs more than 400 surgical cases per year with outstanding results. He is a member of well-recognized organizations, boards, and committees, as well as invited speaker, visiting professor, both nationally in the US and internationally. Dr. Alexandru Mărmureanu, thank you for joining us today. You have the floor.
First of all, thanks for having me. It's a great honor. Actually, it's an honor and a privilege to be here today. And I'd like to thank to the Consulate General of Romania in Los Angeles, mainly Cosmin Dumitrescu is a dear friend, and he's done an amazing work here in Los Angeles. It's much appreciated. He was able to be extremely instrumental in bringing the name of Romanians forward, and it's a great honor to have him here in Los Angeles. Now, Diana Loreta Păun, the presidential advisor, I'd like to thank her very much for helping to set up, and obviously, last but not least, Mihaela Joana, dear friend, her and her brother, I know them for many, many years from Romania, and bună ziua din Los Angeles. Vreau să spun că am avut un pic, m-am gândit complicat. I had a few thoughts in regards to what I should be talking again, because there are a lot of great presentations, very stimulative, I must say, Adrian Bot and everybody else, as well as the rector from Târgu Mureș. My first job after residency was at Târgu Mureș. I have very good memories about that place. Actually, it was Târnăveni and then Târgu Mureș. You know, I got promoted. So it's a great camaraderie, great group of people. I'm extremely proud of being Romanian. I feel totally privileged growing up in a country like Romania and having a chance to practice in the States. And I have to tell you, like all of you guys, thinking outside of the box helped me tremendously in my surgical practice. Now my presentation, what I want to do, I want to put a bunch of pictures and videos, keeping a bit more entertaining in such a way that combine the science with my daily activities. So I want you guys to see what I do every day. And obviously, the end of the presentation, which I understand shouldn't be too long, I truly believe that a good talk is a short talk. Go ahead and ask me any questions, or you know how to reach me on social media and so on. All those things being said, let me share the screen with you guys and see how we can get this thing going. So basically, what are we going to talk about? Minimally invasive cardiac surgery. It's heart surgery, it's lung surgery, minimally invasive. We call it here maximal torture. The less invasive, the more difficult it is for me as a surgeon. Now, this is the UCLA Medical Center. I don't know if you can see my cursor, but my office is here somewhere, Suite 410. And I've been here for 20 years. I train in New York, NYU, New York University, Mount Sinai. 2000, I came to UCLA, and I'm private practice. So, like I said, a good talk is a short talk. That's me skiing. We're gonna move really quick. Again, memories from Predel to Sinaia to Poiana, and I'm still doing it. You know, it's I truly believe that a doctor, surgeon should be well-rounded. And when I don't ski, that's what I do. I'm in surgery, and those are my surgical loops. You see, they're probably 2.5 to 3.5 power magnification. You see there is a headlight. It's not a coal miner. Lumina e la miner, dar de fapt este, it's there to see you in the chest. You see I'm making an incision on the right side of the patient. This guy here is taking the vein for what's called CABG. And CABG nu e varză. CABG stands for coronary artery bypass grafting. So it's open heart surgery. You see there's a whole team here. Like Adrian Bot said very well, it's a team approach. I got probably 10, 20 people in each hospital. I go to 6 hospitals, and that's the way it should be done. Now, this is after I opened the chest. Actually, this is the end of the operation. You can see this is a vein graft going to the right coronary. The heart is what's called a heart-lung machine, the bypass machine. This is an aortic cannula coming at the top, and at the bottom is called a venous cannula. Cosmin, can you see my cursor?
Yes.
Okay.
I can see your cursor. Yes, it's black. It should have been red, but we can follow that.
So this is this is the heart, and the view is from the top of the patient. That's the way the anesthesiologist looks at the patient. This is my right hand. I'm here on the side. On the right, patient stands on the right. The chest is open. And you see what I do to have a motionless field. In other words, I mean, we can give this talk in 2 hours, but it's going to be 20 minutes today. So we have to rush it a little bit. But the bottom line is that all the blood is taken out of the patient through this venous cannula, this tube that goes to a heart-lung machine, and is getting back to the patient through here. This is an aortic cannula. This is the heart. So I can actually do my work on the heart. And this is the view from the right side. This is what I'm looking at. This tube here, what it does, gives potassium. Potassium is like poison. It stops the heart from moving. The reason I'm saying that, because we do this every day the same way. And when I first moved here, I said, my comment was a bit cocky, you know, like, we said, well, every day the same way, no progress. Well, if we do it every day the same way, where is the progress? Well, we do have some progress in terms of now we operate on a beating heart. Basically, let me go to the next slide. And now you see the heart beating. That's the heart back there. And what you hear me, you actually see some of the grafts in here. And the memory graft is an artery taken from the chest wall placed on the heart. And this is called a Doppler. What it does, it verifies the sound that there is blood flow through those grafts. A sort of, you want that sound. You want to be sure that you did a good job, and obviously the patient, it's doing better. Clearly, it looks somewhat barbarian. Probably, if you guys figured out by now, the chest is open. There is a big retractor in here. The question is always, can we do better than this? Now, this is the gold standard. So we're going to go in a few options that was very instrumentative in helping developing. The truth is nothing wrong with this the way it is, but can we get it better? Minimal invasive. Minimal invasive, one of the classifications, first of all, having the patient on a heart-lung machine is not physiologic. In other words, basically, you don't need those tubing and the heparin and everything else. Patients don't tend to like this. So if you can put them, if you can do the surgery without having them on a heart-lung machine, you're better off. They're better off. Beating heart. Now, second, less invasive is smaller incision. You saw those big incisions here. You're looking at that, you know, it's probably a 30 centimeter incision here. Less invasive, less of an incision, yeah? So one of the classification is, where do we go from here? Well, mini incision, direct vision. So you saw that sternotomy back there. So the option would be now to do the same thing through a smaller incision, much smaller, doable. You're not going to be able to do 3 or 4 bypasses, but probably 1 or 2. With a small incision, patient tolerate better, faster return back to work, they recover better. So that's one way to do it. Next thing is direct vision. Do you remember those big loops, ocular lupe? You saw the headlight. That's the way I'm looking. The next part is video assisted. What do? You assist yourself with a video camera. In other words, you put a video camera and you see whatever you can see through your incision, smaller incision, I must say. And whatever you can see, you have a video camera. Interesting, this video camera has got a 30-degree angle. So it's not like this. So what you do, you see around the corners. In other words, it's much easier to see angles and you move it in your hand. The next part would be, the next step up would be robotic surgery, which allows you many degrees of freedom. Now, small incision, direct vision. Micro incision, video assisted. Port incision, very small, video directed. What I don't that anymore. And we're going to go through those slides. I do all the operations looking at the screen, looking at TV, just the way you look at your soccer game. I look at the TV, that's how I do my operations. Next part, level 4, port incisions, robotic instruments, video directed. What does it mean? I tell people, why do I make big incisions? You saw that's the anatomy. Look at me, I got big hands, okay? I got to put my hands in the chest. Now, if I don't have to put my hands in the chest, much smaller incision. So the robotic device has 2 metal wrists, small. They imitate the motion of the human wrist, and they go in the chest. So you're basically outside of the patient chest. You're inside the operating room, but not touching the patient. And the robotic device is inside of the patient and does the work the way you're doing it. So that would be level 4. Now, do you remember that big incision up here? That's actually mitral valve repair, replacement. See, it's a small incision right below the nipple. It's called a mini thoracotomy. So again, it's not up here, it's here. It's much smaller, yeah? So this is actually the surgeon looking into the chest. You see this? Smaller retractor, you see the heart back here. The problem is the heart is not in the right chest. The heart is basically in the middle. So you see there are a bunch of sutures that I place in to bring the pericardium, and that's where the heart leaves, in the pericardial sac, toward the right side. You bring it towards you. Well, what do they say? Muhammad comes to mountain, yeah? So basically you bring it here and you see better. Now, how much can I see through this small incision? Not that well. You see, I have the loops, I have the light, I have a video camera. This video camera is a 5mm, and I don't want to take too much time. We're actually a few minutes into this conversation, but this is actually a robotic arm, you see? So there is a video camera in here. It's called a 30-degree. You see around the corners. I have it in. So how do? I look inside through here, and I look up on this screen. This is an older video. You see the screens are a bit older, but the idea is the same. That's a funny story, is that this is a robotic arm. Initially, we used to have a resident, a fellow, some sort of person holding the camera, and usually they fall asleep because they've been on call all night. So they shake all the time. So you can't really have a stable platform. So we start working this robotic device that holds the camera and it's voice activated. You see, I have a microphone and I say in, out, up, down, left, right. The problem is at that time, the Dragon, the voice recognition system wasn't that good. And obviously I have a Romanian accent and it's got a little trouble. So you keep trying there and sometimes doesn't work that well doing what you're telling. Then we tried to invent a device to put a cursor in right underneath the headlight. In such a way I move my head forward, backward, left, right, the robotic device will do it. Again, this is not, doesn't have the wrist, it just holds the camera. So the problem, we kept moving our hands in the UR like this, so it looked a bit crazy, and the camera still didn't do that well. So we kind of got rid of this, and we moved to, this is a better picture, we moved to different softwares, but the idea is still there. You can see the heart through here. As the initial steps go into robotic surgery. Now you see a little bit better. There is still a retractor in here. The heart is coming up. This is the video camera. As you can see, at some point we got rid of the robotic device. This is just a platform, stable platform. What's the issue here? You still have what's called a thoracotomy. It's much smaller incision between the ribs. The 3D visualization, the way we look at it is 3D, three-dimensional, tridimensional. When you look on the screen, you see only 2D. So you would like to look 3D all the time, because when you place your instrument in the chest, the problem is you need to know how deep you can go. So 3D is definitely helpful. Small incisions, they're limiting range of motion. So it's not much that you can do, but the most interesting part is called a fulcrum effect. In other words, when you put your instruments in the chest, you see I have to move the tip to the right. I'm sorry, the handle goes to the right, the tip moves to the left. So if that's the chest, it's a bit of counterintuitive motion. So in a pure robotic procedures, you don't have that. So we train our residents the step learning curve to basically master this motion. Not to mention, it's the same way like signing your name, holding the pen up here. It's not as easy because you're used to hold it here. So it's all a steep learning curve. Now, you're going to see here next what I'm seeing when I'm in surgery, and that's a mitral valve repair. So you can see the mitral valve, which is the valve in between the left atrium and the left ventricle. It's defective. I'm fixing it. And you can see I'm putting a repair ring. So we can do a replacement, we can do a repair. This is the repair ring dropped into the valve. This is called a nut pusher, goes in and basically secures the repair. Just to brag about how smart the Romanian-Americans are, I actually have a patent on this, on changing the shape of this ring, because what happened is you place the ring, close the patient up, 6 months after, there is still a leak in here somewhere, just because the ventricle remodels. So whatever was good now, 6 months to a year, it changes the shape and you can have a leak. So I have a patent on using radio frequency ablation and actually putting wires in here and changing the shape of the ring to eliminate the leaks, the mitral valve leaks, regurgitation, without having to go in and operate. Now, this is not happening yet. This is just an idea. We still have a lot more work to do on it. Going back to small incision, now you see there is an incision here. That's the mini thoracotomy. The head of the patient is here. There are a bunch of trocars are called. And that's where you duck, is called the robotic device. Those are those sutures that earlier were bringing the heart toward me so I can see better. This is the camera port. Camera goes here. The arms, the left arm and the right arm, those are robotic devices going here. That's how I'm going to do the operation. I'm going to look through here. We'll also inject carbon dioxide, CO2. And that's basically what happened in the opera room. First of all, everybody, like Romania, they're professors. Everybody's an expert. This is the surgeon doing the operation. Outside, you see, it's not touching. The patient is here. Those are the big robotic arms. This guy is called, he's a bedside surgeon. He looks in through a small incision to be sure you want somebody at the bedside, nothing goes bad. There are screens. The surgeon, again, does not touch the patient. We actually done a case from Mount Sinai from New York, was a lab colleague, and the patient was in France. So you have 6 to 8 feet, you have 2-3 meters in between. What stops you from having 6 miles or 60 miles? So this device is the robotic device, has been designed initially for the Army. And imagine that the patient is, let's say, in Afghanistan, even though the Americans are pulling out now, and the surgeon is in Los Angeles. So the paramedics place the arms in Afghanistan, and you can go and click into your robotic device in Los Angeles, do the surgery. And you see everybody, again, it's an expert, wow, put a stitch here, put a stitch there. So you have an audience now. Let me get rid of this. New technology. Now, it's not easy to solve with those robotic devices on a beating heart and special insight. So in the interest of time, I'll go a bit quicker. Those are magnets, yeah? So you see, this is an artery, it's called the mammary artery that gets placed into the heart. This is the heart here. So you have magnets here, magnets to the heart, magnets to the artery. So instead of hand sewing the anastomosis, you actually just click it and stays there. That's one issue. Second, actually that's the way it's being done. That's the artery on the heart. Those are the robotic wrists coming in, and you see the magnets up and down being applied to the artery. So this is the LAD on the heart. So the mammary artery comes in and clicks into that area right here. And you see the way the robotic arms, those are, so my hands are moving and the robotics arms are moving the same way. Even more interesting, it's very hard now if you have a sort of raw motion, you do this. It's fairly easy, but you know, when you sew, the sutures are so thin, you can barely see them with the naked eye. So what we do is like you dial the radio in your car. So we decrease. So in other words, if I move my hand 10 centimeters, the end effector, the tip here, will move 1 centimeter. So you can dial down your motion so to make it more precise. Otherwise, it's very difficult to do this job. This is robotic. We're not going to go much. It's called AFib. I have a bunch of slides. We're not going to spend much time on fibrillation. That's the way you treat it. We do ablation. So that's how we ablate the pulmonary veins, putting catheters around and using a robotic device for the dissection. Epicardial leads, same thing. It's just sort of like a you know, for a pacemaker, the robotic disarm, you can see it. They imitate your human hand motion and they get placed on top of the heart. We can also do this with thoracoscopic, basically, instruments, and I'm going to get there again at the time. This is, again, the same motion. We're going back to the open surgery, and you see a little video. This is a cross clamp, and I'm not sure if you can see very well. This is a dissection. This gentleman aorta tore. The whole thing tear. And you see there is a graft placed up here. I replaced the whole ascending aorta. Young guy, by the way, trauma, car accident, the whole thing tore apart. He's done very well. And you can see the graft up here. Now, again, what can you do? How do we do better? It's very important. You see the incisions are quite small, no more median sternotomy. You have the small incision. It's very important, actually, where do you place your ports? Your ports, how do you get in with your thoracoscopic instruments or robotic device? And that shows you in order basically to be able to get to the correct vessels and harvest everything. So we do this in a minimal invasive way. We harvest the vessel, then it's a small thoracotomy, and the anastomosis is done on a beating heart this time, the old way, no heart-like machine. And those are actually pictures from surgery, from the operating room. And again, we don't talk about the median sternotomy anymore. It's all about minimal invasive, and that's a chest tube. This is probably 1 or 2 days hospital stay after heart surgery. And those are some of the instruments you see, because the heart is beating inside. As you can see, well, you can't see very well, but trust me on that. This is it's interesting. We worked to develop this device. Initially, this area is being placed on top of the beating heart, and the vessel that you're sewing, your anastomosis, is right here. And the way we started, think it was more than 25 years ago, I actually took a fork and I cut the middle tooth of the fork. And I had a resident pushing in with the fork with no middle tooth, so I can show the anastomosis quick on a beating heart. And we're giving a medication to slow down the heart, because if he beats like this, it's crazy. It's fast. You have to be really fast. But if you slow it down, it's a bit easier. So now we don't use a fork anymore. We use this device that's actually compressing the heart. This is called here a starfish, but what it is, it's a suction device that gets the tip of the heart and moves it around, that allows you to access the vessel. You see the difference. This is a median sternotomy. Nothing wrong with this guy. He's going to deal well. Other the incision is not that great. He's got some keloid, versus a mini thoracotomy in here. This is me in surgery, and that's how I look in the OR. And then I'll show you how I look outside the OR. There's a bit of a difference, but that's what I do every day. You see again the loops and the headlight, and you know, obviously the patient. The next step is called VATS, Video Assisted Thoracic Surgery. And what that means, instead of doing those big incisions, we're looking at TV. Look at it. This is the right lung. Actually, this was done last night. I got out of surgery at 12 o'clock, and I want to show you guys some fresh pictures. If you can look at it, there is the video camera right in here. There's no incision. This is the upper lobe, middle lobe, lower lobe. This is the lung. So the whole operation is being done while I'm looking on TV. I'm not looking anymore inside the patient. Yeah. All right. So I am on the basically left side up. Patient is facing the other way, and that's the way we do it. And that's a different view, sort of panoramic. I have a few screens. This is the anesthesiologist, no incisions anymore, right upper lobe, middle lobe, lower lobe. As you can see, the visualization is quite good. I like this picture. It's an older picture, but what I'm looking at, you see, I don't have any sort of glasses, loops, lights, and I'm looking to the chest. How do I do this? With the video camera. So that's our buffer. That's what we use. This is, by the way, it's called a pleurodesis. People with cancer, they accumulate fluid. We drain it. We put some powder in. We scrape the pleura so the fluid doesn't come back. This is the same thing, thoracoscopic epicardial leads placement. The heart is beating, open the pericardium, be sure you don't put a hole into anything. And interesting enough, those leads actually, they're being screwed up, Kushurubel leads. We have a way to screw them into the heart, and you're looking at it. Changing gears a little bit, you know, this is, again, doing a Piatra Craiului. It's me and my girlfriend climbing Matterhorn here. We're going to move to a different topic. So again, that when we don't do surgery, we actually try to play a lot of sports. And changing gears, really quick, I don't want to take much of your time. Ablation or atrial fibrillation. Atrial fibrillation is a huge problem in the States and in Romania, all over the world. And in the interest of time, quick, what we do, there are pulmonary veins on the right side, on the left side, coming to the heart. So if that's the heart, pulmonary veins, right, left, you will have to have a device, a clamp that ablates, and this is the clamp. Allow us to use radiofrequency ablation energy in order to interrupt the signal from coming from each side toward the heart and create atrial fibrillation. So the treatment would be, again, permanent isolation of pulmonary vein, and that's the way it's being done. And those are those ganglionic plexi on the right and left. What they do, they send the signal to the heart. Heart fibrillates, not a good thing to do or to have. You need basically anticoagulation, complication, 1 to 3% per year, so on and so on. People don't function properly. So if and when our colleagues from electrophysiology, they do the same thing going through the heart with wires, they're not successful, that's the next thing we do. Radiofrequency ablation through a minimal invasive approach. Okay, this is the same thing, how we do it. We map pre-op, we map post-op, and then we document that we've done a good job. The left part of the heart, the left atrium, has almost like an ear. It's called an appendage. And that area creates clot in there. And that's the biggest problem. The clot from there goes to the brain and creates a stroke. So what I do, you see it here, there is an instrument like a scissor that cuts and staples, cuts and closes at the same time. On a beating heart, you actually resect that area. So as you can see here, there are no more clots because the whole thing is gone. You also do this, you take a deep breath because you hope your staples are working well. Otherwise, you look at the inside of the heart, and that's not a good thing to look at. Some of the complications, you can have a lot of complications, but it's a good procedure, and we do it quite often. Changing gears again. I like to race cars. As you can see, this is Pikes Peak. You can't afford to have complication. I encourage you guys to read about Pikes Peak. I actually played in a movie. There's a heart surgeon doing this race. Este capa transfăgărășan, but they have no guardrail. So if you somehow miss a turn, you land in a different state, in a different area. So it has to be precise. It's all about hand-eye coordination. Talking about complication, being careful, this is what's called a pericardial window. You see there is a hole in the pericardial sac. I made this hole, by the way, because there is a lot of fluid here compressing the heart, and the heart is not beating. This was a longer video, but I cut it. You see all this fluid. The fluid is what it does, actually. It's, again, compressing the heart, not letting the heart beat properly. So you put this hole in here, and basically they'll tend to do better. I'm toward the end, and we also do vascular surgery. This is the aorta, okay? Those are the head vessels going up. This is called the arch, thoracic aorta, abdominal aorta. We frequently put stents here. We used to do everything open. We still do it open, but we put a lot of stents in here. So the reason I got this is that it's going to be hard for you guys to see, but when people have kidney failure, they put what's called a dialysis catheter to dialyze them, and I'm sure you're familiar with that. Unfortunately, this patient, it's a very sort of crazy case, interesting. The dialysis catheter should be placed into the neck, into the vein, into the groin, anywhere. This time they put it not even in the carotid artery, it was placed straight into the aortic arch and into the aorta. So it's quite a problem. You see the video, this is the catheter coming into the aorta. I mean, this patient can die anytime, can exsanguinate. You see it's a tube that goes in. He should have been in a vein. It's into the aorta, going toward the heart, quite a sizable hole. So those are things that I do every day. You have to figure out a way to take it out minimally invasive or open, and be sure that the patient does well. This is a better view. This is actually a pacemaker coming from the left side toward the heart, and this is the catheter who missed a vein, missed that's what's called the innominate artery, got straight into the aortic arch. Straight. It shouldn't be here. That's supposed to be in a vein. It's in the aorta going toward the heart. So I had to take the thing out. That's how I look when I'm not in surgery. And that's my girlfriend, Laura. So I'm involved in a lot of charity events, and we do a lot of charity in Hollywood and Los Angeles, and that's a night out in town. And let me see what else I got. I told you it's going to be a quick talk. It wasn't that quick, but thank you again. And I appreciate you inviting me, and I look forward to your questions. Thank you, guys.
Thank you, Dr. Marmureanu. I must say that I'm a little bit speechless. I knew the level of your professional level and what you're doing, and I read a lot about your work, and I must confess that I'm a fan.
I was just thinking, did I lose you guys? You're speechless, or we lost connection? I mean, I couldn't hear anything.
I was speechless, but now I'm speaking up. And I hope that you can hear me. Can you hear me?
I can hear very well, guys. I really appreciate you, and again, thank you. And it's just, I'm so always happy to be invited and keep in touch with my Romanian friends, and it's a great opportunity, and I hope you do it again, because again, it's a good opportunity for all of us to keep in touch and hopefully share the information and help the new generation.
Yes, Alex, we will certainly do that again, and the intention of the Consulate General of Romania is to organize smaller events and putting together universities from Romania and students and professors from Romania with you, and not only with you, Alex, but with you, Romanian-American specialists here. And we are very grateful that you already agreed to get into contact with them and to teach them many of your techniques. That's one of the solutions for Romanian research, and we highly appreciate that.
Well,
I must say, Alex, that it was very interesting to see how technology is used and how automation is used in performing modern surgery, heart surgery, and thoracic surgery in general. And that gives me the opportunity to pass on to the next speaker. That is not an MD. He's a physicist. As a matter of fact, the next two speakers are going to be two physicists that are very well involved with their research and their professions and teaching methods into the medicine. So let me introduce to you Dr. Radu Moldovan, and we will continue, Alex, we will continue the dialogue in the Q&A section, because I'm sure that many of the people in the audience would like to ask you questions. There are a lot of doctors there. I'm not going to ask you any questions because I'm a PhD, but in a whole different field. So, but there are.
Let me say, sorry to interrupt you. I mean, it's not a good thing to interrupt the Consulate General of Romania, but I will. I want to give a lot of credit. I just got a text message from Dr. Azam Fire, and I want to thank you very much from Târgu Mureș. I look forward to coming back to Târgu Mureș and operate with you guys. But I do want to, you know, I practice in probably the best centers in the world. I mean, you talk about NYU, Mount Sinai, UCLA, but I want to give credit to the Romanian School of Medicine. I would not have been here today if it wouldn't have been, my parents are physicians. My sister is a doctor. I train at Craiova. And I must say that going to medical school in Romania, I had a lot of role models and mentors. Professor Petre Georgescu was the dean. Dan Mogos, general surgeon, Sherban. I always wanted to be like those guys. Unfortunately, I have to tell you, there is nobody in America I want to be like. I think they want to be like me, but I couldn't find a role model and a mentor here the way I found in Romania. And Romanians are very, very good doctors. They're very good surgeons. They're very skilled. They're smart. I would like to reemphasize that I would like to come to Romania, but I don't feel that I necessarily need to teach. Those guys are very, very good. What I think we need to do is collaborate and cooperate and again, give them credit for who they are, because they're very good. Perhaps with better technology would be perhaps a better job. Technology would help a little bit more, but let it be understood that they're very good doctors. The school is out there. And again, I'm very proud that I'm part of it. Thank you.
Thank you, Alex. Nobody could put it better than you have just done. Thank you for your kind words. It's real what you are telling us. And yes, Romania has an amazing school of medicine, and not only. And we can see that everywhere in the world with our good professionals that work everywhere. And I'm sure that your expertise and your contribution in teaching young students in Romania is highly valued. And I'm very grateful that you agreed to come to Romania, or at least to virtually make some courses for university students in Romania. Coming back to our symposium and passing to the next speaker. So Dr. Moldovan is a biophysicist running the Advanced Light Microscopy core at the University of Colorado Anschutz Medical Campus. His imaging center provides advanced imaging techniques like Nobel Prize-winning super-resolution methods, STED, PALM, and STORM, fluorescence correlations techniques, RCS, RICS, as well as multiphoton lifetime and intravital microscopy. He received his PhD degree in physics from the University of Pittsburgh in 2006 for doing biophysics research on virus infection mechanism of a bacterial host at population level, including arms race modeling. After his doctorate degree, he worked in the Carnegie Mellon University from 2006 to 2010 as postdoctoral researcher studying amyloid beta formation within Alzheimer's disease, employing advanced microscopy and spectroscopy techniques. In 2010, he accepts an offer for establishing an advanced imaging center at the University of Colorado School of Medicine. His collaboration expands beyond Ansho's medical campus, reaching other universities across the US, as well as biotech industry in Colorado. His microscopy and imaging expertise has been used in multiple different biomedical research areas, from neuroscience to pharmacology and cancer research. So we can see how the work of a physicist helps both surgery and the research work of doctors like Adrian Bod. Dr. Radu Moldovan, thank you for accepting our invitations. Thank you for being with us. You have the floor.
So thank you for the kind introduction. Thank you for inviting me to give a talk in such a great Romanian-American event. I'm speechless after the talk we just experienced all together from Dr. Murmureanu, and I have to say the whole event so far been very educational and very cool. I have to tell you guys, my talk will be very different from what you saw so far, but hopefully you'll enjoy it. It'll be very technical, but let's see. Can you see my screen?
Yes. Okay, we can see that.
All right. So yeah, as I said, today I'm going to talk about fluorescent correlation methods in biomedical research. It's a very niche topic, but it does help a lot of research areas from neuroscience all the way to cancer research or other biomedical research. So this is the core, the center I'm running. It's called Advanced Light Microscopy. It's under a bigger entity, the Neurotechnological Center, which holds other sharing instrumentation research centers. And I have a couple of helpers here, as you can see. And I want to mention, I have also a Romanian student who's also watching the symposium right now, Carol Miritza. Hey, Carol. This is our webpage with different kind of instrumentation we have for doing research, everything from confocal microscopy to super resolution and label-free imaging techniques. I'll dive directly into the outline. I'm trying to be short. I'm trying to be on time. So I'll start with talking about the diffraction limited resolution confocal microscopy challenges, and then I'll continue with a little bit about super resolution imaging advantages, and at the end I'll talk with my favorite topics, spectroscopy methods like fluorescent correlation and raster imaging correlation spectroscopy. So let's see. So I hope most of the audience here know about microscopes in general. A microscope which uses light. The workhorse in microscopy is what we call laser scanning confocal microscope, and here is a very brief schematic of it, where you have a laser which is focused by an objective onto your sample. I hope everyone can see my cursor, so you can see what I'm pointing at. So the sample being labeled with fluorescent markers will get excited and emit a fluorescent signal, which will be later detected to a pinhole and get to the image as you raster your sample with this laser and the whole setup. Now the whole idea about the big deal about confocal is that the imaging is detected to a pinhole, so you get only the signal from the very focal plane of your sample. You don't see anything above or below the focal plane, which is a benefit of confocal microscopy. However, this techniques is not giving you the resolution you need in doing biomedical research, and that's just because the light itself cannot be focused in a focal point tighter than, let's say, 200 nanometers, as you can see in this table here, based on the wavelength and the optics you're using. So keep that in mind. In this talk, I'm trying to tell you the challenges or the shortcoming we're having in imaging with light, with lasers, and then what we as scientific community figure out to do in order to overcome those shortcomings. Here is it's a short slide with the fluorescent markers which we use in imaging, and I cannot stress enough this fluorescent marker make our life easier. As you can see, this little mouse here is expressing green fluorescent protein in its skin, so we can easily image any organelles or any tiny fraction of this mouse. Someone got some Roger got Nobel Prize in 2008 for coming up with green fluorescent protein. Now, as I said, the laser light cannot be focused in a tighter point than 200-300 nanometers. For those who doesn't know how big that is, I can tell you right now, that's probably like 500 times smaller than a piece of hair. But this is still not good enough for observing accurately proteins which are in the size of few nanometers. So, for example, here, if you look at the dendritic synapse, like a neuron, you can see the focal point is covering a lot of the synaptic or the dendritic spine, and it's hard to tell if there's any interaction between those few proteins. So we have to come up with a sharper tool than that. So, for example, in these images here, we have a confocal image in two channels. One is showing the actin, the other one is showing vinculin. When you do the super imposing of the two channels, you can see yellow, which means you have in the same pixels both actin and vinculin. My question here is, by seeing that, do we really guarantee that there is an interaction between these two proteins, or it's just that they're sitting in the same 200 nanometer pixel? And that is the challenge we're having, or we were having last century, for trying to figure out the interaction between molecular intracellular entities with the imaging techniques. So it seems like this fight of battling the diffraction limit resolution barrier got very, very strong, and in 2014, 3 great scientists got Nobel Prize for breaking this barrier by coming up with super resolution microscopy techniques. I want to mention the guy in the middle here, Stefan Hell, was born in Romania. He moved to West Germany when he was a teenager. I met Stefan in 2012 before he got his Nobel Prize, and I remember speaking to him in Romanian, and he told me his mom was still speaking to him in Romanian, and he understands Romanian, but he doesn't feel like he can speak it well, so he wouldn't speak. So I thought that's a good parenthesis for this talk. So his Stefan Hell idea about breaking the barrier, the resolution barrier, was to superimpose a another laser beam, which was focused in the shape of a donut, as you can see up here, and this extra laser to be high power, and pretty much they stimulate all the particles, the fluorescent particles in the donut. So therefore, you'll be left with a smaller effective light volume, which is shown in here. So here is an example of images, confocal images and STED images done in our imaging center. As you can tell, on the left, the confocal image, which uses the standard diffraction limit resolution, is much poorer than the STED image on the right. The resolution is improved like four times, and definitely this would help in seeing better what's happening in that neuron. But the question is it good enough to see the interaction between two proteins which are much smaller than, again, this new better pixel resolution, which is in this case like 50 nanometers. The other method which got Nobel Prize was the single molecule localization microscopy, Palmer Storm. In this method, to be like quick, by using chemistry tricks on these fluorescent markers in your sample, you make them blink, and when you take like many thousands of pictures, each picture will catch only a subset of those fluorescent markers being on, and because it's just such a small number of subset molecules, you'll be able to super localize them, as you can see in here. So you end up with a super resolution map of your image, which indeed is great. It's like 10 times better than confocal. The pixel size here is like 20 nanometers, but still not good enough for probing directly interaction between proteins, which are much smaller than 20 nanometers. And this is an example also taken in our imaging center. You can see up here is the standard image, and then down here you can see the palm super resolution map. So definitely you can see much better with this super resolution technique, 10 times better, I would say, but again, not good enough for molecular interaction in live cells, because first of all, this technique is very slow, right? It takes few minutes to take that many images to build up such a super resolution map. So now the question is, can we measure molecular dynamics and interactions within the diffraction limited focal point? And the answer is yes, by measuring fluorescent fluctuations. For example, sorry, I need to drink water. So, so for example, in this confocal image, if you park your laser spot in this area of the cell, and you're watching the fluorescent signal, you will get something like this in time, right? This is the data. And now, if you try to correlate the fluctuations of this signal with itself tau time later, like sometime later, you'll get what we call autocorrelation function. So the autocorrelation function looks something like this. This is real data. I'll get back to the other slide in a second. I think I swapped the slides in here. So this is the real data, very rich in information. You can get anything from like concentration of the molecules in your light volume to the size of the molecules and diffusion of the molecules, and so on and so forth. And here is the models we're using to fit those autocorrelation function and data. And obviously, because we're doing live cell research, we We have the three-dimensional diffusion model available, as well as a two-dimensional diffusion model where you look at the protein diffusion within a membrane. Then we also have a one-dimensional diffusion model as well, and even an anomalous diffusion model, which is very encountered in real-life cells, because the proteins diffusion in cells can be confined to volumes of your light volume. So we have all the models needed for this in order to figure out the diffusion of the proteins we're observing, as well as their radius, so on and so forth. Now, that's very good, but we're still not to the point where we can beat the super resolution imaging with this technique. And why is that? Because we didn't talk about the molecular interaction yet. So this is the setup which will enable us to do these fluorescent correlation techniques. Actually, in this case, it's cross correlation, because we're trying to correlate the fluctuations of one channel with the fluctuation of the other channel and see if there's any correlation in there. So what you're going to see in your light volume, presumably this light volume is seen from above. That's why it looks like a circle, but it's still that tight focal volume we're used to by now. So what you see here, you see the two different proteins. One is, let's say, labeled with red, one with green. And then what we do, we look at the correlation between these two channels, and we'll end up seeing only the pairs. So again, this is a cartoon. You do not get to see the actual molecules, the actual pairs of molecules. What you get to see is just light, just signal. But doing the correlation of these two channels, by doing the correlation of these two channels with each other, different times tau, you'll be able to build up a cross correlation function, which will tell you if these pairs are moving together or not. For example, to make this understandable by everyone, just imagine you have a red light on your head, your friend has a green light on your head. From the helicopter, from the chopper cannot really see you individually. It can see only the light where you are. And we're in the same room. Obviously, the helicopter will not be able to tell if you two guys are interacting, if you're next to each other, or if you're separate in different corners of the room, because the chopper sees only the signal, only red coming from that room. But if the chopper is looking, if the observer from the chopper is looking at the intensity fluctuations of the green and red light coming and exiting the room, it can tell if you and your friend coming in and out of the room together or not. So this is pretty much how we can tell for sure if we have binding in this live cell experiment. So for example, and this will be a quick example. This is a classic paper for our campus. It's coming from Lab, and back then they were trying to figure out the sodium phosphate co-transport for NaPi2A and NaPi2C. They were expecting both of them to be interacting with each other on their way up the microvilli. You can see here, these are the kidney cells, and they have this brush border membrane microvilli. So they were expecting to see these two NaPi2A and NaPi2C traveling together up the microvilli, just because they're both regulated by the same hormone, the PTH hormone, parathyroid hormone. But the data informed that the data didn't show that being the case, and you can see it right here. So the blue cross correlation curve, it doesn't look like there's any cross correlation between these two channels. Interestingly enough, NaPi2A is interacting with SHANK2E, a different protein, which they didn't expect, and they learn from these kind of experiments. And you can see the cross correlation. It's obviously above the zero line, meaning there is cross correlation between these two proteins. Now, so far we talked about this fluorescent correlation spectroscopy technique, which involves the cross correlation between the two different channels in time. But we can take this further one step and try to do this while you're rastering your focal volume across your sample, and then you can get also the correlation, the spatial correlation information. And that's exactly what the raster imaging correlation spectroscopy technique is about. So now the autocorrelation function will look like a surface. Before it was just a curve. Now it's a surface because it has two variables. It's spatial correlation and temporal correlation, which you're considering in this method. And here you have some examples as we measuring these. And this is another example from the same lab. They were trying to figure out the diffusion of NaPi2a and nerve proteins and see if they move together. And obviously you can see that with the previous techniques, with the fluorescent correlation spectroscopy, but you also can see it with raster imaging correlation technique, which takes into account the spatial correlation. And here is the proof. So you can see this is the cross correlation data for those two proteins, NaPi2a and NRF1. And I think, I get to my last slide here. And I want to think I convinced you that fluorescent correlation methods can provide answers on molecular interactions better than confocal and super resolution microscopy. And also I want to remind you that in biomedical research, relevant imaging data is not always just pretty pictures. It can take other formats, as for example, this autocorrelation function we're talking about today. And thank you for your attention, and I'm looking forward to hear question for this at the end of the session. Thank you.
Thank you, Dr. Moldovan. A very interesting presentation with a very particular approach of science, and showing us how interdisciplinarity between sciences can contribute essentially to breakthroughs. And we could see the cross-correlation in medicine, in microscopy here from your presentation. And I will remind the people that are now attending this symposium and that attended also the symposium of last month that was about the space sciences, how spectroscopy is used for studying the universe, the outer space, and how people use that in order to plan strategies for bringing resources from outer space on Earth. So it seems that physics has no limits. So
it's everywhere for sure.
And I'm sure that our colleagues that are attending now this meeting are looking forward for the Q&A session, for asking questions about your presentation, and we'll stay in this interdisciplinary environment, and we pass to the next speaker, one of your colleagues, Dr. Remus Oșan, and he is also a physicist, and he accepted our invitation to participate as a speaker in the symposium of today, and I thank him for that. I'm trying to pull out his presentation here in order to be able to introduce him to you. I'm sure that, yes. So allow me to say to tell you some facts about Dr. Remus Oșan. He is currently an associate director and for data science at Gilead Sciences. So he's more or less a colleague of of Dr. Adrian Bot. And this is the same company that provided the remdesivir, but I'm sure that he's going to tell us about these details shortly. So originally, he is from Baia Mare in Romania, and Dr. Remus Oșan holds BS in physics, a bachelor in physics from the University of Bucharest, and a master in computer sciences and a PhD in physics from the University of Pittsburgh. Dr. Oșan has pursued postdoctoral studies in molecular biology and neuroscience at Princeton University and mathematical biology at Boston University. During his postdoctoral studies, he has developed statistical and classification methods for large neural population activity and discovered new encoding features and consolidation of episodic memories in the brain. Dr. Oșan then took an assistant professor position in the Department of Mathematics and Statistics at Georgia State University, where he established a mathematical biology and computational neuroscience research lab focusing on research on neuroscience, epilepsy, olfaction, neural growth, cancer, and heart dynamics. Dr. Oșan has obtained federal NSF and NIH, American Cancer Society, and industry funding award and industry funding, and has received and has received an NSF Early Career Award at the Mathematical Biosciences Institute. In addition to advising PhD and masterants and the undergraduate students at Georgia State University, Dr. Oșan has led the study abroad program in mathematical biology in Cluj-Napoca in Romania from 2014 to 2016. In recent years, Dr. Oșan has transitioned to a career in the industry by first working at a couple of startups where he led an analysis program for biological biomarkers used for monitoring multiple sclerosis patients and selected a set of biomarkers to be used for actionable insight into disease progression and therapy. And he led projects on cancer imaging data with a focus on tumor classification, segmentation, and evolution, and on telecommunication data with a focus on system monitoring, evolution, and anomaly detection. As Associate Director in Data Science at Gilead Sciences, Dr. Oșan is currently leading the data science efforts in predictive analytics for supply chain, machine learning, and scientific computing algorithms in process chemistry and natural language processing methods, and also in data mining techniques for large-scale databases. Dr. Remus Oșan, thank you again for accepting our invitation. We are looking forward to learn from your presentation. You have the floor.
Thank you. Thank you for the nice presentation, which I wrote myself, I guess. So that's always good. Okay. So today I'm going to talk about some emerging data science approaches in pharmaceutical and biomedical research. And I sort of want to give a complimentary view from my colleagues. We had some amazing talks where we discussed some transformative technologies, such as the CAR-T. We saw some real life and death videos of surgery, also new advances in spectroscopy, the state of medical research and collaborations in Romania. I want to focus a little bit about what happens when you get a hands on the data, what is the approach. And as we know, there's an invasion of big data nowadays. We have too much data, and what do we do with it? So this is a slides that I had a collaboration with GSpace, which was featured in one of the previous symposium on space technologies. And some of the abstract ideas are similar in the terms that we need to have a focus on what do we do with the data. First of all, we need to acquire and to store it. Then we need to do some data cleaning. We need to process it a little bit. And then we can go into deep analysis and machine learning methods to uncover the hidden patterns that exist in the data. And this is mainly where I will be focusing today. And then obviously, very important as well, communicate the results of the data to the management and build up roadmaps and strategy. That is also very important. So interestingly enough, there's been an evolution. What has been thought to be distinct path between computer science, visualization, and statistics and mathematics now are converging. This data science is an all-encompassing idea on how we process the data, what type of models do we use, what type of data mining techniques. So I want to talk about, first of all, a case study, which is called the API camera. And this is a methodology for looking at some properties for the active pharmaceutical ingredients, such as particle size, surface area, and blood flow. And the idea is that we want to look at these properties together to understand if a tablet becomes really useful for this solution and an effective treatment. I think my slides, for some reason, are going a bit back and forth, so I apologize for that. Essentially, the reason why we look into this was to troubleshoot ideas that arose during drug development. So I don't want to go into a lot of details, but it's easy to understand that there's a correlation between surface area and particle size, morphology, surface texture, and porosity. So why this issue is important? Let's assume that we're looking at multiple dimensions, and I want to try to keep things mathematical, but also a little bit intuitive. So one would think that you see this linear relationship and your products are homogeneous. However, if you look at different dimensions, such as the surface area versus the particle size, you realize that there's actually some heterogeneity, meaning that there is a lot of variability. So how do we go about uncovering the patterns that exist in here? We notice, for example, that if we have an assumption that this variability comes from the fact that we have two types of classes, now we see that we obtain much better results in terms of doing some tight fits of the function. So you sort of have to think of, can I draw a function naturally between these lines, just for the more wider audience today? So why is this relevant? Well, this is relevant because it turns out that people who are doing the manufacturing build products that have similar properties. And obviously, some of them are going to be higher quality, some of them are going to be lower quality. And you can get this global view by examining things in 2 dimensions, which is the left panel, or in multiple dimensions, which is the right panel here in 3D. And this movie just demystifies a little bit how one goes about finding this quality of the fit. Initially, you put all the data points into 2 categories randomly, and then one by one, you try to switch the membership from one class to another and go through all of them and see if the quality of the fit improves. And if it does, then that's a new iteration, a new starting point for a new iteration, and then you try to move points between 2 different classes in such a way that every single time you're actually improving the quality of the fit. So as you can see in this particular application, eventually the quality of the fit becomes quite good, and then things become fairly intuitive in the sense of producing this intuitive separation between these 2 classes, which is essentially the left panel here. But I thought that it was interesting to show the process, because sometimes they say, machine learning, mathematics, statistics, artificial intelligence. A lot of this stuff has some intuitive ideas that I think can be explained fairly easily. In conclusion, for this case study, we use these machine learning techniques to understand the methodology and then to determine the best partition to figuring out how to understand and troubleshoot issues that arose doing manufacturing. The second case, I'm just giving in the sense that we want to show that some of the things are also some simple statistics. So we want to understand the development of predictive biomarkers. These are very important for detecting the early stage of the cancer based on some biomarkers. Some cancers are more difficult than the other in the sense that they're missing some of these biomarkers. So often there is a case for developing new prognostication and predictive biomarkers. So here's an example of a case why this is very important. If you are looking at recurrence between African American and European American patients, you can conduct studies showing that African Americans are actually more prone to have late detection, and they're also more affected and they die in larger numbers than European American. Another example of machine learning techniques and computer visions applied to these systems is this biopsies, where you look at potential tumors and then you need to identify them using computer vision and then use that information to also look at differences between these two populations that I mentioned, African American and white. Finally, here's an example of an algorithm that I developed in terms of a new biomarker. It is called Centrosomal Amplification Score, CAS. And I just want to provide some intuition in terms of some images. Essentially, we have these centrosomes, and these centrosomes can differ in numbers, and they also can differ in size. So an example of high severity in here on the left is you have a lot of centrosomes. These are these red cells, and that has high severity. And if you compare to low severity, there's a smaller number of them. But there's also a structural amplification, meaning that they come together and they become larger in size. So as a result, this also causes a problem. And if you put these two quantities in a score, you can come up with a mathematical formula that gives you a new tool to aid the decision making and improve treatment recommendations. And you can actually show that this is relevant in the sense that patients with higher CAS are more likely to experience cancer recurrence, and also they die at an earlier stage in comparison with cancer patients that don't have high CAS. So this was a collaboration with Dr. Renasterea at Georgia State University, and we have 2 patents for showing that this prevalence, frequency, and severity can be described by these new biomarkers called CAS. This led to a number of papers in which I was the lead for the statistical analysis and models. In interest of the time, I'm going to move quickly on another case, which I hope is going to be quite intuitive. Brain study functions, essentially recordings from the regions of the brain in normal and mutant mice. And here is again an issue of collecting the data, doing signal processing, collecting databases. The design of experiments is essentially some mice which are being dropped, the cage is being shaken, they have some loud noises. In essence, some stimuli that are likely to bring about large activity patterns in different regions of the brain. So this is the setup and the recordings. Then you get this signal and it has to be processed and filtered, and then eventually have some population recordings which look like this. So hopefully you see the movie. The cage on the left is going to be dropped, the cage on the right is going to be shaken, and the mouse in the middle is going to receive an air path. And the things that are moving from left to right are the activity patterns. Whenever a spike occurs in a neuron, digital signal is being propagated from one region to another, and then you get a lot of cross-talking between these regions of the brain. So then the difficulty is to look at these large activity patterns and quantify what has happened. How are these activity different patterns between stimuli A versus B versus C? And then we came up with some statistical methods related to dimensionality reduction, such as principal component analysis and multiple discriminant analysis, that allows us to compress those large activity patterns into a simpler representation into 3 different clusters. These are baseline activities. These are activities related to the dropping and the shaking and the air path at the population level. Now, what's really interesting, if you see the movie, is that not only get these activity patterns when the stimuli is occurring, but also later on, without being prompted, you're going to see some activity patterns that are very reminiscent of what happened earlier. So this is a visual representation, sort of like a triangle, and later on, the same triangle occurs with a smaller magnitude. So we think that these are some reverberation of the memory traces. The brain is thinking, what happened? How can I prevent it? How do I understand what's going on? And this essentially allows us to do monitoring of the network level activity patterns in the brain. So if you look at it visually, these original events are larger magnitude. They sort of look like triangles, and they're distinct from different type of activities, drop, shake versus air. And these activity patterns happen in the same plane, and they look like smaller triangles. And we think that these are essentially telling us something about timing of the reactivations and re-experiencing the pattern. We also conducted some studies in which we varied the intensity of the stimuli. And what we have discovered was actually very interesting. A lot of things that are involved in remembering things are not that highly modulated by the intensity. So in other words, this mouse here is remembering that there was a drop, but the fact that there was a drop from a lower height or a higher height later on is not being played with such intensity. It's more like a feeling, oh, there was a drop and the magnitude of the intensity is a bit smaller. So this also has resulted in a number of papers being published, and the main idea is that we can show that there are some coding units that contribute to real-time activity patterns of the memory, and this internal representation of the external events is not by recording all details of the experiment, but sort of the most important, the most salient ones that later on are used to generate a more global, a more generic representation of the kind of events that has happened, and then that forms the basis of processing things and remembering them for a long time. So I wanted to be fairly brief because I know that we are short on time, but I just wanted to give you this idea about data science is having a huge impact nowadays in pharma and in biosciences. Essentially, as I mentioned, big data is coming. We're going to be swept by the sea of data, and we need to understand what's going on. There's cloud solutions like Amazon Web Services and Google Cloud that are taking care of that burden, so people don't actually have to worry too much about storing things. But a big need remains in terms of having qualified people to understand the data, to provide data analysis and modeling, the machine learning and artificial intelligence type of models, and then essentially not only creating black box model, but being able to explain why this is the case. And why do we have a result versus another? And also connecting a little bit with some of the previous presentation, also creating models about understanding how cancer works. What are the models of the brain that work? So not just having everything in terms of computing correlation between all possible quantities, but also having models of genetic cascades that happen in the into the gene patterns and into the neuron patterns, and I think that's also very important. So this is part of the industry collaborators. So these are the acknowledgement. This part here was essentially for the API camera was in collaboration with my colleague Henry Morrison from Gilead. The part with the cancer research was in collaboration with Dr. Renasterea from Georgia State University and a lot of graduate students and some other colleagues. And then finally, also the neuroscience collaboration, mainly done in part with Joe Chen at Princeton University and at Boston University. So with that, I think, let me stop the sharing. I do have sort of some unusual acknowledgements. When Dr. Dumitrescu, I'm going to call you doctor because you said you have a PhD as well, said April 28th. So April 28th is my brother's birthday. So happy birthday, my brother, who's also on the call. And also my parents are very happy to see me at conferences, but nowadays virtual is the way to go. So my mom and my dad, thank you very much. One of the other things that was very interesting, it's a small word. As Dr. Dumitrescu connected us, I realized that, yes, Dr. Bot was a colleague of mine from Gilead, so that was very interesting. The first thing that I remember is that Dr. Radu Molovan, his name is actually Bixi, was a colleague of mine from the PhD program, so that was very nice. And then I was talking to Dr. Zamfira, and I realized him being at Târgu Mureș, he probably knows my uncle, who's a neurologist there. And he said, yes, he's a colleague and a friend of his. So all of a sudden, out of a group of 7 speakers, I have 3 very close connections, which were very interesting. So I want to really thank Consul General Domnul Dumitrescu for putting us together and connecting us. I think we get to be focused a lot on our research and about relationships at our work and in our immediate group of friends in the diaspora. But we also need to remember the connection with our colleagues in Romania and keep having them and keep strengthening them. So for that, thank the organizer for this. I think this was really important. And with that, I think that concludes my presentation.
Thank you, Dr. Roshan. I'm so happy and so grateful at the same time to see that all of you keep a very strong connection with your roots, not only professionally or academically, but also as good Romanians. As a matter of fact, one of the objectives that Consulate General of Romania in Los Angeles has with this symposia is that of setting up a network with Romanian-American researchers, and not only Romanian-American, Romanian and Romanian origin researchers all over the world. And I'm sure that there is a high interest in setting up such a network. And with your support, I'm sure that we will be able to connect most of the Romanian researchers, not only from Romania and the United States, but also from Europe and from other countries in Asia. This is one of our objectives, and I'm sure that we are going to fulfill that eventually. Your presentation has been really, really interesting, and you suddenly raised even higher the level of interdisciplinarity and the connection, the amazing connection between these sciences and how medicine could benefit from that. And the link that you mentioned of your presentation is not only with the previous ones from this conference, from this symposium, but also with the next presentation of Dr. Caius Radu. He is going to somehow sum up elements from all the presentations that we had until now, and because he's going to tell us about novel approaches to image and target nucleotide metabolism in autoimmune disorders and cancer. And I can see that practically we have here in the title elements from all the others presentations. But first of all, allow me to introduce Dr. Radu, Caius Radu. Fine professional and also a dear friend of mine, is professor at the Department of Molecular and Medical Pharmacology and Surgery at University of California, Los Angeles. He also serves as a vice chairman of the Department of Molecular and Medical Pharmacology and co-director of the Cancer Molecular Imaging, Nanotechnology, and Radiation Research Program. Comprehensive Cancer Center at the UCLA. He is also a co-founder of Sochi Biosciences and Theta Corporation. Professor Radu received his MD diploma from the University of Medicine, Craiova, Romania, and completed his postdoctoral training in immunology and cancer biology at the University of Texas Southern Medical Center in Dallas and at the UCLA. His group aims to advance the understanding of fundamental biological processes at the interface between metabolic, signal transduction, and immune networks in cancer and other diseases, and to leverage his knowledge towards the development of new diagnostic and therapeutic approaches. Additional areas of focus include studies of novel mechanisms that regulate nucleotide metabolism in cancer and immune cells, and the development of nucleoside imaging probes as non-invasive biomarkers of immune activation. The potential of the program is demonstrated by recent work which uncovered a new role for the nucleoside salvage pathway in hematopoiesis, identified a metabolic kinase as a new therapeutic target, developed new PET probes for nucleotide metabolism, several of which have been successfully translated to the clinic, and discovered novel small molecule inhibitors of deoxy. Oh, I think that Dr. Radu is going to help me with that.
Exciting.
From which a clinical candidate compound with a with a low nanomolecular affinity and oral bioavailability has emerged, with indications in selected cancers and autoimmune disorders. Oh my God, I don't have my glasses, and it is really difficult for us law school graduates only to pronounce the things that these medical doctors are doing. So thank you for your existence. Dr. Caius Radu, thank you again for joining us and for accepting our invitation. You have the floor.
Thank you, Your Excellency, for this very kind introduction. I think you did actually great at pronouncing the majority of those terms. I also like to thank you. You are the driving force behind this initiative and great many others. I want to thank you for the opportunity to present at this important symposium. I'm extremely honored to be in the presence of very distinguished guests and colleagues, and I hope that today's presentation will and discussions will lead to impactful collaborations. Let me try to share my slides.
Great.
I want to start by giving a very brief overview of the department that I'm part of, which is the Department of Molecular and Medical Pharmacology at UCLA. In many ways, this department is unique at UCLA, given that it consists of over 40 faculty who have interest not only in fundamental biology, but also translational medicine. And the concept that at the core of the department is something that we call a triangle concept, something that we actually have developed in our lab. And the idea is that one has to innovate in each of the corners of this triangle in terms of discovering new aspects of fundamental biology. Then you have to also develop new ways to measure biology. And we heard quite a few examples, amazing examples today. And once you have done those two things, you can now take them, combine them together, and develop new ways to actually treat disease. I'm going to give you a brief example of how we try to do that in my lab. And again, being in an academic lab, it's very different than being in a company, especially a big company like Gilead. So I think that the audience will actually benefit from seeing both perspectives. My lab is, I'm trained as an immunologist, and my lab is studying the immunological roles of molecules called nucleotides, which are essentially form the basic blocks of life. And what we all know that they form, they are essential components of nucleic acids, but what we are more interested in, actually, it's the fact that they have very important effects on the immune system. And here, what the slide is showing that some nucleosides, actually, such as adenosine, exert very strong immunosuppressive effects, where others listed on the right, such as guanosine and this kind of dinucleotide called cGAMP. And one nucleotide, the nucleoside that I'm going to talk about today, called deoxycytidine, they actually now have effects that lead to immune activation. And that will be the focus of the brief presentation today. In the light of following what has been discussed today, I strongly believe that it is critically important to build partnerships between industry and academia. One cannot simply stay in the academic ivory tower any longer. It's becoming unattainable. So I realized that essentially all of our current projects in my lab involve academic industry collaborations. Those on the left, I'm listing some of them are with companies, most of them in the immunotherapy field, such as Arcus Biosciences and Amgen. But others kind of like Aquitas, they are involved in formulating mRNA vaccines that essentially the lipid nanoparticles used in the COVID-19 vaccines that most of us have now received. Today, I'm going to focus actually on projects that relate to two other companies that I'm a co-founder of. One of them is called Tritera. And Tritera is a company that focuses on developing novel therapeutics, and the second one is Sophie Biosciences, which is actually more on the biomarker diagnostic side. A key component of our research toolbox in the lab is a technique called positron emission tomography, or PET. I guess many of you, all of you probably heard about PET, is now, it's impossible to imagine a hospital anywhere in the world that doesn't have a PET scanner. But there is a lot more to PET that just what can now be done in a hospital, and that is because this technology, it's essentially a platform in which one can take a variety of molecules, starting from small molecules and then increasing the size to peptides, antibodies, antibody fragments, even nanoparticles. They can be labeled with these special radioisotopes which decay by positron emission. I'm listing several of them here. And then these probes are now injected in a living organism in very small amounts, nanomolar amounts. And they can be then detected using a principle of coincidence detection in a PET scanner, such as the one that I'm showing you here. And the technology is fully scalable from humans down to experimental animals such as mice. And what you get up in the end, it's a quantitative out of a PET scanner. It's an image that it's usually whole body, and then it's a three-dimensional image and can be fully quantified. What drew our attention to PET was the critical need for new treatment response biomarkers, especially in the fields of cancer and autoimmune disorders. And what the slide is showing here, that for a patient affected by a malignancy or by an autoimmune disorder, usually now they will receive these therapies that are, in the case of cancer, will be more and more often based on training, reprogramming the immune system to fight these tumors, including immune checkpoint blockade, adoptive cell transfer therapies, as we heard from Dr. Bo today, cancer vaccines, and various combinations. In the case of an autoimmune disorder, of course, the patient will receive an immunosuppressive drug, such as dexamethasone, for instance. But in both cases, what we need to have are treatment response biomarkers that can rapidly identify patients who respond to treatment or those who do not respond. Now, these biomarkers can be of can come from many sources, and including blood biomarkers, sometimes biopsies. But what we were interested in developing, and that's what PET can actually enable, is essentially developing whole body non-invasive imaging biomarkers. What I'm showing you here is a strategy that we have developed to identify new imaging probes that are sensitive to changes in nucleotide metabolism, and therefore be able to identify activated immune cells at the whole body level. As you can see here, the process resembles in many ways the one that the process used to develop new drugs. You have to identify a target, then you have to find a probe that can bind to the target, and then this is a very iterative process in which you have to make a number of modifications. And then once you have candidates, you have to validate them first in animal models, and we use mostly mice, and then take the most successful ones and test them in humans. So I'm showing you here a slide that summarizes a lot of work that took actually more than 10 years. We had to develop a number of these imaging probes. Each of them, it's a significant challenge given that the half-life of a radioisotope like fluorine-18, it's only 2 hours. So then you have to have very good chemists who work with you, who can actually attach this fluorine-18 to a molecule and in that time frame. And sometimes that requires a number of synthetic steps, this is a major challenge. Then you can produce these probes eventually, and you have to evaluate them and learn from each one of them and continue to improve them. So for us, it took about 3 generations of development. In each case, we had multiple candidates to have a probe that actually functions well in humans. Going back to the mouse data, what can these probes do? What I'm showing you here on the left, it's a PET scan of a healthy mouse and contrasting with it a PET scan of a mouse that actually has now an early stage autoimmune disorder. And what you can immediately see are these kind of strong signals in the cervical lymph nodes, which are a reflection of this autoimmune process. Now, what we can do, we can take the same mouse and scan it again after 3 days of treatment, and as you can see here, these abnormal signals completely disappear. We can do the same now in a different type of autoimmune disorder, which is a model for a human autoimmune disease called multiple sclerosis, a very severe disease and quite widespread. And as I'm showing you here, that before disease is induced in mice, the brain of these mice appears essentially almost completely devoid of the probe being accumulated. When mice develop the disease, we can see accumulation of the probe in the brain. And after treatment with an immunosuppressive drug, then we actually see the signal disappearing, and that coincides with mice actually now recovering from the disease. Maybe even more importantly, we have taken these probes in humans, and this is a patient from my colleague and friend at UCLA, Tony Ribas. What I'm showing you here is a scan of a patient before immunotherapy, and this patient has a melanoma lesion in the lung. You cannot see it with this probe because the probe is more specific for immune cells than tumor cells. However, after immunotherapy, the same patient scanned with this probe. Now we can detect increased uptake of the probe in lymph nodes and even the appearance of a new lymph node, which reflects a systemic activation of the immune system induced by this immune checkpoint blockade approach. Now, in biology, we always have to learn the mechanism of action. How does these probes work? Again, cutting a long story short, what we can do, once we have a candidate, a suspect, gene that might be involved in this process, we can engineer mice to essentially to delete the gene, and that's exactly what we have done here. The gene that our probe is actually monitoring is called deoxycytidine kinase. It's a metabolic kinase, and its function before the studies was completely unknown. Now we know much more about it. However, kind of what I'm showing you here is how a mouse looks like before the gene was deleted. We can then treat the mice with a compound that will essentially trigger a deletion of this gene at the whole body level. We can scan them again, and you can appreciate almost a black and white difference between the two scans, indicating that our probe has high specificity for the target. We also learned in the course of these studies that mice in which this gene has been deleted, they are fully protected against an autoimmune disorder that I mentioned before called experimental autoimmune encephalomyelitis, which is a model of the human disease multiple sclerosis. Essentially, what I'm showing you here is a clinical disease score that we apply to mice, and mice in which we induce the disease, which are these orange and orange circles and gray squares, they develop quite severe disease. However, mice in which the gene was deleted, which are the blue triangles, are completely protected. Now we also know that these mice have a normal lifespan, so deleting this gene is not detrimental. They are not more susceptible to infections or to development of tumors. And that prompted us to now ask the question whether we can develop an inhibitor of this gene that could be used for treatment of autoimmune disorders and selected malignancies. Again, this slide is summarizing a significant amount of work, starting from what we call in biology a high-throughput screen, in which a chemical space, in this case about 90,000 chemicals, were tested to identify those that can actually inhibit the activity of this kinase called deoxycytidine kinase. Again, this process is quite iterative, so you find candidates, they bind to your target, but not avidly enough. They don't have good drug-like properties. So we had to go through multiple rounds of what is called structure-activity relationship optimization, each of them guided by high-resolution crystal structures of kinase and the drug candidate. Finally, now, again, after 3 of these rounds and then almost probably a total of 200 new compounds, we have 2 clinical candidates, out of which one was selected, and this compound is called PRE515. What can this big compound be used for? One example is shown here. We are looking at a systemic model of acute lymphoblastic leukemia, in which a combination therapy based on this novel BCK inhibitor is being applied. On the left is what we call a Kaplan-Meier curve, looking at survival of mice, and as shown here, mice that are not treated, this gray blackish line, they die rapidly. Sometimes they don't even survive 3 weeks after we induce leukemia in them. But mice that have been treated with this combination, they survive long term, and we don't see any detrimental effects on body weight, which again, it's a good indication of the treatment being well tolerated. So what's next? It was gratifying that at the end of like almost a decade of work developing PET probes, which are the biomarkers for this drug, and these drugs that the company to which UCLA licensed these compounds has now received FDA approval to initiate a phase 1 clinical trial, which we hope to enroll the first patient in the first quarter of this year. And as I mentioned before, the protocol will include not only treating the patients with this drug, but also using PET technology and the PET probes that we have developed to measure whether a drug is engaging the target and inhibiting its activity. So to conclude, I think the take-home message is that it's important to develop not only drugs, but also biomarkers that can quickly tell us whether those drugs actually work. The second lesson is that we have developed a first-in-class drug that targets a metabolic kinase, and this was done entirely in an academic lab. And that is really, again, as mentioned before, this requires a team. So in fact, it requires a team of teams because we had at one point a team formed of biologists, immunologists, and chemists, and essentially working closely together and to develop these compounds. And I guess the most important lesson for us and take-home message is that when I started at UCLA, it was really uncommon to work with, to have industry partners. Now it is uncommon not to work with industry. So as I mentioned before, essentially all our projects in the lab, and they don't involve even imaging, they involve like many times fundamental biology. We work closely with different companies and help them understand how their drugs or therapeutic approaches work and how they can improve their efficacy by developing what I think will be the future of oncology, and that is combination therapies that are rationally designed. That gets me to my final slide, and again, this makes the point that it's a team effort, and we have many collaborators. There is always an important Romanian connection, I guess, for all of us, and those are people are listed here on the right. They impacted my career decades ago when I left Romania, and it was quite an experience to go from Craiova to Dallas, Texas, and I'm very grateful to them. They were my first mentors. And also funding. Life in academia is not easy. It is incredibly difficult, competitive to obtain grants, and that takes a significant amount of our time to write and compete for these grants. And here are our industry partners. And thank you, everybody. And I look forward to the discussion section and questions.
Thank you, Dr. Radu. Thank you for most interesting presentation. As I suspected, a lot of elements that we met in the previous presentation were to be found in your final detailed story and presentation of your work. Congratulations for what you're doing, for all of your achievements in UCLA. We totally agree that the partnership with the private sector, with the industry, is essential for funding research activities, and of course that grant procedures are never enough. And I think that some of our partners in Romania, at the Romanian University, should learn and should make some kind of an import of know-how as how to successfully finalize procedures for funding research activity in Romanian universities. And I'm sure that your contribution is a giving back, thank you, between inverted commas, to the universities in Romania would be very highly appreciated and very important for professors in Romania. And now, for wrapping up our first part of the symposium, I dare say that I'm not only pleased but very happy with the level of the presentations. We had the opportunity of opening the works with a perspective from the social civil society, from an NGO, talking about closing the healthcare gap and health education and prevention. And then we continued with a very precise analysis of the Romanian medical research from Professor Zanfirei, the rector of Târgu Mureș University of Medicine. And then we dove in some futuristic, I dare say, some incredible presentations given by Adrian Bot with curative interventions through immune engineering, continuing with a real adventure. I don't know, it was much better than Chicago Med, with the presentation of Dr. Alexandru Mărmureanu, with the videos that he presented, even the video that he recorded last night with his in one of his interventions, surgical interventions. And then we had two amazing interdisciplinary presentations about how physics, mathematics models are used in medical sciences and biotechnology, with Radu Moldovan from Denver University and Remus Oșan from Gilead. And we close the session of presentation with Caius Radu with these new approaches on imaging and target nucleotide metabolism. I'm going to learn how to pronounce that, Dr. Radu, I promise you that. So before going to the next part of our symposium, I would like to acknowledge, checking out the list of the participants, to acknowledge the participation in our meeting of some representatives from the US Embassy in Bucharest. Thank you guys for joining us and taking our invitation. And also we can see professors from university professors from Romania, but also from, if I'm not wrong, from Manchester. So I'm really happy that this conference, these works are going to have such a huge effect, and they raised such a big interest. And also checking the list of the participants, I would like to acknowledge the presence, and we are very honored to see that in the attendance there is also the Deputy Secretary General of NATO, so Mr. Mutajoan. So thank you, Deputy Secretary General, for joining us. I'm sure that your time is really limited. We are very honored. So it's amazing. Thank you. And now I open the table for some free discussions between the speakers, and then of course we are open also for some questions. And seeing that there are a lot of doctors in the attendance, I think that we can take the questions directly, so not necessarily by sending them to me by chat and then repeating them to the audience. So please, if you have any questions, I think that you are already able to unmute yourself and to ask them. So, table is open. Thank you. And for giving the time, yes?
Can I just say how amazing this all was? And I can't help but think that put together everything that you do in a Romanian context would raise the GDP by 3%, at least. It's absolutely awesome, awesome advances. And I'm still trying to figure out how we as the state can match make best your expertise with that of other Romanians in your fields of expertise, either in other countries, but especially also in Romania. And symposia like these, of course, are very helpful, but there's, at least feel the need of something more meaty, more concrete being yielded by these, from these discussions. I'll think about it and get back to you. But I think actually matchmaking is what we ought to do best. Both you have done an amazing job, Cosmin, of finding such talent. And it's our responsibility, I guess, to find your colleagues where in Romania you can share these this expertise. So thank you again. It was absolutely awesome.
Thank you, Sandra. Yes, the Romanian talent, and you're so right. Romanian talent is certainly one of the bases for building and continuing building a very strong academic dimension of the strategic partnership between Romania and the United States. Their contribution is really amazing, and the importance of the data, of the information, of the scientific breakthrough that they are giving the world from here is really, really important, not only for our two countries, but also for, but also globally. So that's why we are trying to build this network, and we are trying to put them together more often and to make them know each other here in America, but also in relation with Europe and of course with our mother country. So that's the purpose of our mission here, and I'm very grateful that the presidential administration and all the institutions in Romania are giving us all the necessary support for that. And I'm also really grateful for what my partner from today, Joanna, made for making this symposium possible. Thank you, Mihaela. Thank you from the bottom of my heart, and congratulations for what you're doing. And I'm asking the first question, if I may, and I invite you all to ask your questions to our speakers today. It's a unique opportunity. So, doctors and professors, what do you think that's the main challenge for medical sciences and for medical professionals after the pandemic of COVID-19?
Can start to take a stab at that if you can hear me first. Can you hear me?
At the end.
And I'll invite my colleagues to do that. And I would like to exemplify with, again, the 4 pillars of an ecosystem that I think it's very, let's say, creative. It's very conducive to creation and acceleration of the innovation cycle. So the 4 pillars, as I explained, consist in the appropriate environment, scientific environment, technical environment, number one. Number two, appropriate financing. Number three, of course, a very permissive, let's say, framework from a legislative standpoint. Number four, a strong IP framework. So these are the key components that I think allowed us to shorten up the innovation cycle. And this is the keyword, to adapt very rapidly to global crisis. And I exemplified how actually discoveries that were latent for a couple of decades, such as DNA-based vaccines or RNA-based vaccination, came to life and fruition during a global crisis, right? We looked at what technologies are the ones that could translate faster to a vaccine that is potent and scalable, rapidly scalable with minimal price. And at the top of the list were RNA-based vaccines, as an example of gene-based programming of cells or living tissues. So I think that in order for us to meet tomorrow's challenge, we need to learn from the lessons actually acquired during this pandemic crisis. And one of them is to reprioritize some of the technologies that we and others have developed and kept on the shelf for decades, and put in place the appropriate processes to accelerate their utilization at the next crisis, be that owing to a pandemic crisis or something different. So that's at least my perspective from where I'm sitting. And of course, I invite my colleagues to comment and add.
Well, if I can add, well, Kaius, you want to go first?
No, Alex, go first.
Well, I just really quick, I fully agree with Adrian, Dr. But remember, team. So in other words, it doesn't matter what those vaccines are. If people don't take them, it's worth nothing. So to me, education, I think it's very, very important. And I'm not talking about Romania. Every day, spend a lot of time in the hospital.
Alex,
I have friends and colleagues that are not vaccinated. So it's going to be very hard after the pandemic to go back to normal if you still have COVID around. So I think the biggest problem would be vaccination, to convince people. And by no means I'm saying don't force anybody. They have freedom and don't want to do it. They shouldn't be getting it, period. However, they need to understand that it's very hard to restart and reintegrate in a lot of structures. And I'm talking about hospitals, nurses' care, nursing homes, etc., without being fully vaccinated. So to me, that would be, at least that's what we're dealing with right now. And after this, there are a lot of other issues, but that's the most important part, I think.
I completely agree with both Adrian and Alex, and maybe thinking about it from the point of view of what are the roadblocks. One of the roadblocks that of course, funding is always a roadblock, but sometimes you actually have enough funding, and it's actually difficult to start a clinical trial that uses a mechanism that we call investigator-initiated clinical trial. So finding ways to speed up translating technologies or new therapies developed in the academic environment to industry and get them evaluated into clinical trials will be remains a process which is often too slow, and we know now that it can be much faster. It's incredible that essentially in a year, less than a year, we had 2 mRNA vaccines, essentially first in class, being developed, pushed through clinical trials, and now being used to save a huge number of lives. The other kind of aspects, roadblocks, are sometimes difficult to anticipate. Now there is actually apparently a shortage of pipette tips created by the pandemic that can affect, essentially not only academic labs but industry as well. So I think that what we have to do to be better prepared for these kind of situations in which things deviate from normal in a very significant way, be more proactive. This pandemic is the first one in 100 years, but I don't think we'll have another 100 years before the next one occurs.
I think I can also add another component, so that's not directly related to the research, but is the way we live our life. We've been impacted by the pandemics in terms of personal life and in terms of work. And one thing that's probably going to be different when things go back to normal is the way we conduct work for the people who don't need to come up to the lab or to do experiments or don't go to the hospital. In particular, talking about data science, there's companies such as Facebook that now are moving to a model that says you can work remotely for the rest of the employment, meaning that you don't have to be on site in the Bay Area, you can be anywhere in the US or potentially abroad. So on one hand, it opens up more possibility for collaboration via the virtual tools. On the other hand, there's this aspect of we are not built to have no social interaction and sit in front of a computer. Typically, we like to be social and talk, have lunch, and that's when you get a lot of creative ideas. So that's another aspect of how we have been impacted, and it's clear how and when things are going to go back to normal.
I think we have a question from the attendants from Romania. Dr. Bogdan Bazga, please. He's an expert in security, so he's not an MD, so I'm expecting something interdisciplinary.
Thank you very much for this amazing conference. Thank you, Cosmin, for organizing this very important event, especially in this period. And I'm more than honored that I'm Romanian when I see my Romanians in US, as I was also proud last month when it was another conference on space. I was honored last year to took part in the special conference organized by the Romanian presidency, especially by Miss Diana Păun, presidential counselor, dedicated on this issue. Besides of that, in the last year, it was a report on the OECD, especially dedicated for the antimicrobial resistance, where it was, could I say, put the red light for all over the world on the use of antibiotics. It was also included the use of antibiotics for animals, and they explained the fact that quite could arrive to 30% of the antibiotics to the people which eat in practice that food from animals and and my question, sorry, to the doctors, how much influence for you, for your work, for the medicine, this unlimited use of antibiotics? Thank you very much.
So let me rephrase that, if I may. So I think Bogdan is asking us how much does the overuse of antibiotics on food, no, animals and plants influence your work. What's the extent of that influence, and how can that be reduced?
Dr. Marmureanu, would you like to take a stab, and as an immunologist, perhaps we'll follow you? Can't hear you.
Dr. Marmureanu, you should unmute yourself.
I'm sorry, I was on the phone at the hospital. I didn't hear the question. If you don't mind repeating the question.
The influence in our daily life of overuse of antibiotics coming from foods, as well as usually abuse of excessive utilization of antibiotics in our field. And I believe you can go first, and then we'll continue with some other perspectives, if that's fine.
Well, I think in our field, I think there is to a certain degree, there's excessive utilization of everything, antibiotics including. I mean, you want to talk about CAT scans, you want to talk about labs, you want to talk about God knows what. I mean, you know, even surgical procedures. Almost everybody I know between 30 to 50, they had surgery. 20, 30 years ago, everybody does. However, going to antibiotics, yes, they're overutilized. And what we decided to do, at least in my practice, we get an ID, infectious disease specialist, that's going to basically guide the medical management. But again, that comes down to education, comes down to people feeling comfortable in their daily lives with what needs to be done next. So with all those things being said, I'll let the next speaker go on.
I can continue if that's fine, and I definitely invite Dr. Radu to weigh in, as he's an expert also in inflammatory diseases, imaging those T cells. But from my perspective, where I'm sitting, there are two categories of problems with overutilization of antibiotics throughout actually food, nutrition in general, and other means. The problems are as follows. The modification or alteration of what we call the microbiome, which is the collection of all bacteria at the interface between our internal organs, internal milieu, and the external environment, including the gut. The microbiome actually controls, based on data accumulating during the last 10 years or so, the tonus of the immune system, right? So if the tonus of the immune system is changed or altered, then there are two categories of problems that may arise. One of them dealing with innate immune surveillance in general against infection and cancer. And the second category of problems, actually inflammatory disorders, which also sit at the interface between innate immunity and organ-specific damage. So I see that overutilization of antibiotics can actually lead to problems in those areas, as well as actually the effectiveness of immune intervention in, for example, cancer. So I'll stop at this, and perhaps I'll invite Professor Radu or others to comment.
I could not agree more. This was actually a very good question, given that that we haven't talked about how important the microbiome is for responses, not only disease pathogenesis, but responses to therapies. From this point of view, we are probably only at the beginning of understanding how excessive uses of antibiotics will impact the microbiome. I also would like to advise against using excessive probiotics, that you can find them now everywhere, because their effect on the microbiome, the gut microbiome, it's really not well understood, and could paradoxically lead to resistance to various therapies. And of course, then there is like excessive use of antibiotics in animals, where kind of like the enormous amount of antibiotics being used that way creates a risk of essentially kind of now put selective pressure on pathogens, and again we are going to end up with a limited spectrum of antibiotics that can be deployed when we actually need them. So the incidence of this kind of like antibiotic resistant germs is increasing, and yeah, it's a major issue. I don't know exactly what can be done about it. We keep talking about it for decades, and the problem becomes more and more severe rather than being solved.
Dr. Dumitrescu, may I say something also?
Please.
Yeah, good afternoon and good evening. I'm Ilana Costea. I have been a professor at university in Los Angeles area. It's not a PhD offering university, it's a primarily teaching university, California State University Northridge. What fascinates me, first of all, I want to congratulate you all, you, Dr. Cosmin Dumitrescu, General Counsel, for organizing these sessions, and also the speakers. Of course, I am fascinated by all the speakers because they are fields which I'm not very familiar with. But my question to you, physicians, my question to you, scientists, is what is the way in which you can impart the message of the importance of vaccination to people in a way in which they will understand that it's not something that pharmaceutical companies want to make money. It's not a way in which just doctors want to demonstrate their knowledge. It is really a need. What kind of mean, and what's that mean to educate people in a way in which they absorb this information?
Thank you for your question, Kostya. I would like to start with Dr. Roshan, as it's very late and he needs to leave us for another meeting. Please, Dr. Roshan, can you start, please? You're a representative of the industry, so I'm sure that such a big company as Gilead has a very strong strategy for educating the large public about using some of its products. So I'm sure that you can give us an insight, very well argued.
I'm not sure I want to educate the public all the time about all the things that go in the products. But, you know, again, talking in terms of data science technology, I think you mentioned, for example, neural networks and ethical AI. There are some tools that are being used by experts, and sometimes there's not enough visibility in communicating with the larger public what assumptions go in using them. And often you get into issues like confirmation bias by selecting the training data in a biased way. You create a tool that more and more is going to have an unfair application in the larger world. So we can go into some examples, but I think the only way to prevent these issues is to have a workforce or education system that allows people to become more quantitative and more knowledgeable about the emerging systems, and then that will allow people to have a more critical view on some of these emerging technologies. I hope that couples well with the intent of your question.
Dr. Borg, your work is based on this kind of research and product commercialization.
Yeah.
What do you think about it?
I think we need to basically get ourselves out of the ivory towers once in a while. We're way too busy, way too in our world and enjoying actually the science as well as their products that we understand to a much higher extent than most of the population. So we have to partner up with actually people who are policymakers, people who are in administration, and make sure that we get the word out. It's not sufficient for us to exchange this extraordinary science and it's the fruit of our labor amongst ourselves, but we need to get this done on a wide scale, and we need to find the appropriate ways to doing it. In large pharmaceutical companies, including Kite, Gilead, and so on and so forth, we have cohorts and cohorts of medical science liaisons who are actually their own business each and every day is to educate the medical personnel in hospitals about new products, new treatments, new paradigms. But we have actually to scale up this particular principle, especially for us to achieve the readiness state in the wake of a pandemic event and in expectation of other global crises to be prepared. Thank you.
Marmuranu, what's your opinion on that? Can you hear me? Oh, he's on the phone, so we'll continue. We'll continue with Dr. Radu.
Just in a second, hold on.
Cosmin, I think can I
please
something? I think that Dr. Adrian Bot is right, and I think that the scientists and big pharma and the companies that bring all this advancement could partner with patient groups, with NGOs, with communicators, people that really do that or are experts in that field, and people that can reach a large amount of the population interested. And again, education is very important, I think, starting from kindergarten and at every stage, medical education and people to know how to prevent and how to diagnose early. Thank you.
Well, I'm off the phone, and I'd like to reemphasize Mihaila's point about education, if I may. We say here that little knowledge is worse than no knowledge. In other words, if you know, dacă știi puțin, e mai rău decât n-ai ști deloc, pentru că ei se cred savanți acum, profesori. So I think education, it's very important. And I don't want to say anything bad, but we have those issues here in regards to people that they're not following the party line and the rules and so on. So I fully agree with Mihaela.
I'm not sure if I can see a hand raised by Professor Daphne Mureșanu. I'm not sure.
Okay. I have a question. Can you hear me?
Yes.
Yeah.
If you can ask who. Okay.
Yeah, it's Adriana Friedman. I'm from Memorial Sloan Kettering in New York. It's more a comment and just voicing again what actually Dr. Radu from UCLA, Caius Radu said regarding partnering with academia. So I think 7 years ago, I was very naive, and I was trying to establish a collaboration with Professor Maia Simonescu at the institute in Bucharest, which is a very famous Institute of Vasculature Diseases, and Adrian Mania, and we wrote together a grant, which we submitted to the NIH. And I was naive enough thinking that we're going to get funded. And we weren't funded. And unfortunately, that collaboration stopped right there. And I'm very sad about it, because I would have loved to work with these people. That institute was established by the late Professor George Palade, just across the street at Rockefeller. So I was very naive then. Now I know that that's what I have to do. But now I want to ask our surgeon, from Los Angeles. I don't, at the end of the day, killer number one is really heart disease, and I don't know of any companies that will fund what we wanted to do. We know today that for our cancer patients, now we have survivors that 10 years after those survivors will suffer from cardiovascular disease, be it from radiation therapy, be it from chemotherapy, and there isn't much we can do for them. Is there any company that I can start again to work with this institute in Bucharest, which I would love to work with, and try to do something about these patients? Any suggestion?
Are you asking me?
Yes, you.
Yeah. Well, I
ask you because you're a surgeon, but still, I think you're the most qualified. I think, unless anybody else jump in, please jump in.
Listen, I like to think I'm qualified, and I like to think for the vote of confidence, but I think probably Mihaela, Joanna is more qualified than I am because they raise more money. This is an ongoing problem. This is an ongoing issue, lack of funds and for different research. So it's an ongoing competition. I can tell you this is a well-known issue, and it's coming from New York to LA to Romania.
Yeah, nobody in particular, as you know, a lot of
well, I mean, I can tell you that, first of all, Memorial Sloan Kettering is a premier organization and a great center. And I think probably you should reach to the foundation. And at the end of the day, you got to go and present your work and find some donors that would be able to support a project. It's not an easy task, and I wish you best of luck with that.
Yeah, there is competition everywhere, but that's what I'm saying. I'm just bringing it about because I started this in a long time ago with Maya and Adrian Mania, and it got stuck. Now I know where I'm supposed to go, but still I cannot identify one company that will be interested in this.
I think it's a dual responsibility, if I may. I think you have to find the appropriate people in here, and then perhaps our general counsel can help in Romania. Perhaps they can do part of the job. I'm sure there are people that are be willing to step in.
Okay,
And,
so that's why I brought it up.
the way to do it.
So I brought it up because I would love to work with the institute in Nicolae and Maia Simeonescu in Bucharest. I would love to work with them. It's a great group. I would love to work with them. But okay, we'll try. We'll keep trying.
Another idea is there's a lot of funding for research and for this type of activities at EU level. I'm not sure if they found cooperation with US entities, but I think that for this financial exercise, they're open to cooperation outside of EU. It's Horizon projects that actually the Romanian part could look at.
Okay, we should.
Dr. Haimovic, if I may, allow me to add two things to what Mihaela Joana have just said. You've just mentioned that 7 years ago you wanted to start this cooperation.
We wrote the grant, yeah. We wrote the grant together, yeah, sorry.
Yes, to bring the grant together. There are two separate segments here that you should consider. First, to bring a third party, so a third partner in your project, maybe adding the University of Medicine and Pharmacy from Bucharest,
fine,
institute, and then apply for funding for EU funds on a Horizon program, EU program that Mihaela Joana has just mentioned, would be a good thing. 7 years ago was a very different period in Romania, a very different one. So a lot of other opportunities for funding open, especially from the EU, from the European Union.
And I'm happy to hear that.
And I'm sure that a good project is going to be financed. If you need help with contacts, we will be happy to help you. And I'm sure that Dr. Mărmureanu is right. Probably NGOs like Fundația Renasterea of Mihaela Joana, that knows everything in Romania about that, are very useful for
no pressure
for helping you navigate.
Okay.
At the end of the day, the project that you're putting down is the one that gets the grant. So this
also,
is 7 years ago. Please.
Sorry. Can I just chime in? The strategic partnership with the US has a big ambition to develop into the economic and the business area and to get American investors into Romania. What I've heard so far are extraordinary projects of great value, both financial and otherwise. So maybe a way to handle this is to matchmake American corporations, and Adriana, Dr. Haimovic says. Okay, says she tried, but maybe if we understand better the type of corporation, and with the help of our friends at the American Embassy and the economic section of the American Embassy in Bucharest, we can reach out to those US corporations who want to take advantage of the framework of the partnership to invest in Romania by having already the seed talent to build on.
I mean, it can come from there. It can come from the EU, as Mihaela mentioned. I mean, or the Stimatul Consulat. I mean, doesn't matter. I guess as long as we can get funded, both.
And that was the second segment that I was going to mention, the segment that Sandra Plalong mentioned before. But I think that we still have Professor Slevin online?
Hello.
Hello, Professor. You're calling from Manchester, yes, and I think that you've got some information for Dr. Haimovic, if I'm not wrong. You can help right now.
I wanted to make one comment, just a little bit about that conversation that you're having now. And for example, in UK and around some parts of Europe, we have what's called angel investors and investment groups. And they're actually, you know, always looking to support technology and medical advances that are in the middle stage, you know, so they're a bit too early. So the big pharma companies and the large biotech companies are not interested still at this stage, but they're prepared to invest $500,000 or $1 million or something like this into these type of projects. Second thing I wanted to say is that, you know, I have a philosophy, which is that if you really believe you're writing a project which is good and which has the potential to change healthcare, to be translated and transformative, then there must be a way to get it funded. And for using that principle for the last 25 years, probably I've been lucky enough to raise money with a team of people, so a network of collaborators who are all better than me in what they're doing, from all around the world. So, you know, most certainly most of the money that would have been raised for projects hasn't come from UK. And the last thing I just want to say is Professor Azamfire is here, and he's been instrumental in not persuading me, because it wasn't a difficult decision to make, for me to come over to Romania to work in the university, UMFST, where we, apart from being successful in gaining project funding, I believe that there's the necessary expertise and potential and intelligence and intellect, you know, to be successful and possibly be even more successful in pushing forward the boundaries than we are here in Manchester in the UK. So, I mean, I'm really excited about coming over to Romania to try and drive these principles. And this meeting today is just an example which proves to me and shows me that I made the right decision.
We congratulate you for that, Professor. The source and the resource of talent in Romania is really amazing. And this type of cooperation, international collaborations and so on, are a cornerstone and a key for development in any field, science included, any kind of science. If there aren't any other questions, as in Europe, we are pushing the midnight right now, I would like to express my gratitude and to thank each and every one of you for attending this online symposium. I really hope, and we really hope that it met your expectations. And we are looking forward to having you again with us for the next symposia and conferences and scientific communication that we are intending to organize. We will maintain this kind of multidisciplinarity approach, and of course the international environment and participation. I would like also to extend my gratitude to Fundația Renasterea and to my partner Mihaela Joana that helped with organizing this amazing event. I was expecting something very special, but it exceeded any expectations of mine. So thank you very much, everybody. Looking forward to have you again as guests of the Consulate General of Romania and of any other institution of Romania. We are here to support you. We are here to build relations, and we are ready to make everything that's possible for you to be more proficient and more efficient in your work, and of course, in working with Romania. Thank you very much.